Good morning. Hello. I'm not hearing you properly.
Okay. Am I audible now? Hello. Am I audible now, sir? Hello.
Yeah. Now I am
Yeah.
hearing you.
Okay. Okay.
ರವಿ ಆ ಕಂಪ್ಯೂಟರ್ನ ಆ ಕಡೆ ತಿರುಗಿಸ್ತಾರೆ. ಇದನ್ನ ತೆಗ್ಬಿಡಿ ಫುಲ್ ಇದನ್ನ ತೆಗಿದೆ ಅಷ್ಟೇ ರೀಸನ್ ಏನು ಅಂದ್ರೆ ಓಕೆ
Yeah. This is the one. Madam. सर अभी ऑनलाइन है सर को प्रेजेंटेशन ये हो गया गुड मॉर्निंग ऑल ऑफ यू let me introduce our first speaker of the fourth day of workshop. I welcome all of you again on the fourth day. I hope that most of you will be joining us soon. So let me introduce Professor K. Girish sir. He is a professor in Tumkur University since 2015. And he has he he has many a publication, more than 100 publication with total citation of 6,800 and more, with H index of 43 and IPN index of 97. Sir has a very elaborate research career, and he has research articles in many prestigious journals, including Journal of Senior Research, Trends in Biotechnology, Nature Publication, PNAS, Hematologica, GLIA, Free Radical Biology, and many others. And he has, till now, he has guided 6 people, and he has research in venom toxicology, and he has made documentary by Rajya Sabha television, and he has been the external reviewer in many international reviewed journals. For example, Nature Disease Primers, Physiological Reviews, Hematologica, Cell Death and Disease, PLS1, PLS, Neglected Tropical Diseases, Scientific Reports, Journal of Ethanol Pharmacology, Toxicology, Biochemistry, and many others. So, sir will enlighten us with his talk. I welcome sir, and I hope
Thank you,
that
sir.
all our participants will be benefited by your research, and many of them are budding ones, so that they will be delighted to see your research, and also will be guided by you, and will inspired by you in all the efforts. Thank you, sir.
Thank you, sir. Thanks for nice introduction.
Yeah. So yes, sir. Yeah.
Yes,
Your
sir.
slide is visible, sir. Please make into presentation mode. Yes, sir. Now it's in presentation mode. Thank you, sir.
Now it is, you can see my slides now, sir.
Yes, sir. We can.
Yeah. First, I would like to thank Dr. Professor Das, sir, for giving me an opportunity to share our research ideas with you guys. Meantime, I would like to thank Dr. Nitesh Singh for providing a platform to show our recent research to you guys. In next 45 minutes, I'm going to talk more on neutrophil functions and its restoration by melatonin, a redox modulator. As we all know, immune system is a network of organs, cells, and proteins that defends the body against infectious agents like fungi, bacteria, and viruses, and as well as foreign materials such as toxins, pollutants, carcinogens, and parasites. I'm sorry. Slides are not moving.
Sir, please click on the slide, sir. Then we'll be able to move.
Okay. Sorry.
Yeah.
The immune system is not an organ system, but it is a complex networking system, includes so many organ systems. Like skin is the biggest organ system of our body. It works as a physical barrier in order to prevent the invasion of microbial agents. Next, lining of mucous membrane. If microbes can cross the skin, lining of mucous membrane will prevent the entry of microbes into cell and tissue. It is also working as a second physical barrier. Then many secretions of our body, like tears and mucus, they have lysozyme. It works as antimicrobial agent, thereby it can kill the pathogens. Bone marrow produces platelets as well as WBC. WBCs are phagocytes. Platelets and WBCs are considered as innate immune cells. They help to bundle up the microbe and give the microbe to WBC for phagocytosis. Liver as an organ, it can make more number of complement proteins. These proteins try to opsonize the microbes, and they will aid in the microbicidal effect of immune system. Lymphatic system produces B and T lymphocytes. T lymphocytes secretes more amount of cytokines and chemokines. B lymphocytes can make specific antibodies against specific antigens. Thereby, it helps the immune system to clear the microbes. Most of the tissues of our body have a resident dendritic cells, so they can fight against microbes. Immunity is a state of protection from infectious diseases. This works as a defensive system against invading pathogens, including viruses and bacteria. This system removes senescent and aged cells as well as damaged cells, including it can clear tissue debris. This system identifies identifies and destroys the abnormal as well as mutant cells from the body. This system also rejects the foreign cells as well as tissues. It also induces inappropriate responses against allergens by inducing hypersensitivity reactions. And meantime, sometimes the system raises antibodies against its own tissue. Best example is autoimmune disorders or autoimmune diseases. An immune response is the reaction of immune system to an antigen. It's nothing but immune response. Immune response can be broadly classified into innate immune response and adaptive immune response. Innate immune response is non-specific in nature, and we can consider this as a first line of defense, and it is fast when compared to adaptive immune response. The major cells involved in innate immune response are neutrophils, mast cells, basophils, eosinophils, natural killer cells, macrophages, and dendritic cells. This system don't require any sort of antigen. Then adaptive immune response is considered as second line of defense, and it is highly specific to a particular antigen. It takes long time when compared to innate immune response. T and B lymphocytes are the major cells involved in adaptive immune response. T cells can produce different chemokines and cytokines. Both of them are either pro-inflammatory or anti-inflammatory in nature, depending on the physiological state or pathological state. Then B lymphocytes produces specific antibodies to specific antigen. That's why we can say adaptive immune response is dependent on particular antigen. So the innate immune response can be classified into subcategorized into two things. One is external defenses, another one is internal defenses. External defenses, generally they are anatomical barrier. There are different sort of factors involved in, they can work as a external defenses, which includes mechanical factors, chemical factors, and biological factors. Internal defenses includes both cells and as well as acellular components. When it comes to anatomical barrier, especially mechanical factor, skin serves as a mechanical factor. It won't allow microbes to enter into the body. Then mucociliary escalator, the best example is lung, which is having ciliated cells. That ciliated cells, with the help of mucus, they will try to remove the foreign particles as well as microbes from the lung through mucociliary escalator mechanism. Then flushing action of saliva and tears as well as urine removes most number of pathogens from our body. So these mechanical factors, they will prevent the microbial infection. Then when it comes to chemical factors, sweat is having a lot of antimicrobial peptides, and our stomach is having HCl. The pH of HCl is less than 2. Then tears and saliva, it is having lysozyme enzyme. Then uromodulin protein present in the urine serves as an antimicrobial agent. These things, these chemical factors, they always try to kill the pathogens. That's why we can consider these factors as chemical factors. They work as a external defense system in immune response, in innate immune response. The biological factors includes microbes. Many parts of the body we have normal microflora. In our body we can find more than 1,000 species of bacteria. They always compete with pathogens for nutrients and space. Thereby these normal flora try to, they are not allowing pathogens to grow. So these factors serves as a external defensive system in an innate immune response. When it comes to internal defenses, as I said, blood is the main internal defensive system in innate immune response. When you centrifuge blood in presence of anticoagulant, you will get three different parts. One is plasma. That plasma generally contains less number of platelets, but more amount of complement proteins and coagulation proteins. Both of them are considered as innate immune proteins. Complement proteins, they induce opsonization of microbes. Coagulation proteins, they are involved in coagulation. Then cytokines, depending on their property, either pro-inflammatory or anti-inflammatory, they works against microbes. Then Buffy coat contains WBC and platelets. Both of them are considered as innate immune cells. Platelets generally bundle up the microbe and give that pathogen to the WBC for phagocytosis. WBC itself is a phagocytic cell. It will try to engulf the pathogens and as well as infected cells. So, these are the internal defensive system of innate immune response. These slides represent cells which are involved in innate immune response as well as adaptive immune response. And as I said earlier, adaptive immune response takes days together, whereas innate immune response takes very short time. It happens within hour. The major cells involved in innate immunity are basophils, eosinophils, neutrophils, mast cells, and NK cells. Since my talk is restricted to neutrophil, from now onwards, I am concentrating more on neutrophils. Neutrophils are most abundant cells in the category of WBC, white blood cells. They are granulocytes in nature because they do contain granules in their cytosol. There are three types of granules you can find in neutrophils. One is primary granules, secondary granule, and tertiary granules. The content of each granule will be different. They have more number of antimicrobial agents and hydrolytic enzymes as well as iron binding proteins. So, when neutrophils undergo activation, these granules will undergo degranulation. Neutrophil undergo degranulation. They release the contents of primary, secondary, and tertiary granules. The granular content serves as a they can kill the bacteria and they can digest the bacteria. So, it is having multi-lobed nucleus. That's why we are calling neutrophils as polymorphonuclear cells. They are phagocytic in nature. So, generally neutrophil perform three different functions. One is phagocytosis, another one is degranulation, third one is netosis. When the size of the microbe is smaller than the size of neutrophil, then neutrophil try to engulf the bacteria and forms phagosome. That phagosome fused with lysosome, the entire structure we can call that as phagolysosome. Since lysosome is having hydrolytic enzymes, it can hydrolyze the microbe. So, thereby it can eradicate or eliminate the pathogens. If the size of the bacteria is bigger than the size of neutrophil, in addition, if neutrophil is unable to engulf the bacteria, during such situation what happens, so many neutrophils will sit on the bacteria and they will undergo degranulation, they release their granular content. So, since as I said earlier, so granular content is having lot of hydrolytic enzymes, antimicrobial agents, they will kill the bacteria. If phagocytosis and degranulation won't work in order to remove the pathogen from the system, then neutrophil uses third option, that is known as netosis, neutrophil extracellular traps formation. So, in this event, neutrophil ejects its DNA. The ejected DNA serves work as a spider web and it entrap the bacteria. Since the DNA is decorated with a lot of hydrolytic enzymes and antimicrobial agents, it will try to kill the bacteria. Netosis, there will be two types of netosis. There are two types of netosis. Depending on the situation, neutrophil uses. So, all these three functions, they are dependent on ROS generation. So, ROS play reactive oxygen species play a very crucial role in neutrophil function. From this, we can say reactive oxygen species is very essential as well as detrimental, since it is inducing lot of oxidative stress. So, in order to maintain the RAS, the neutrophil cellular redox system should work in a balanced manner. Otherwise, neutrophil cannot perform or display its function in a proper manner. So, we asked a question, what is the role of redox system in neutrophil function? By asking this question, we designed some experiment to prove our hypothesis. So, the primary and most important redox system of any given cell is glutathione cycle or glutathione system. Glutathione cycle is having two enzymes. One is glutathione peroxidase. It is a selenium containing enzyme. It detoxifies the hydrogen peroxide, and meantime it can convert in other words, meantime GSH or glutathione is get oxidized. Glutathione is a tripeptide. It contains glycine, glutamic acid and cysteine. So, while detoxifying the hydrogen peroxide, the glutathione get oxidized. The oxidized glutathione is highly reactive. It will react with another oxidized glutathione by forming interdisulfide bridges to form GSSG. GSSG is two molecules of oxidized GSH or glutathione. The oxidized glutathione can undergo reduction to reduce glutathione by the enzyme glutathione reductase. Glutathione reductase requires NADPH, uses NADPH as well as FAD as a cofactor. In till now, there are many GPX mimetics you can find. So many selenium containing compounds and selenium based nanomaterial serves as a GPX mimetics. They can work as a glutathione peroxidase, but till date there is no mimetics for glutathione reductase. Generally, glutathione synthesized by two enzymes. One is gamma glutamyl cysteine synthase. This enzyme combines glutamic acid and cysteine to form gamma glutamyl cysteine. Then glycine will add to the gamma glutamyl cysteine to form tripeptide glutathione by the enzyme GSH synthetase. The levels of GSH modulate the enzyme action of gamma glutamyl cysteine synthase by feedback mechanism. This is the most essential redox cycle in any given cell. It is true with neutrophil also. So, in 2013, there was a paper, glutathione reductase is essential for Ward's defense against bacterial infection. When you inhibit the glutathione reductase enzyme in neutrophils unable to unable to display its basic function like phagocytosis and degranulation. Thereby, everybody came to know that glutathione play a very critical role in maintaining the glutathione cycle for normal functioning of neutrophils. So, there are many reports like as I said earlier in 2013 Ann et al. Demonstrated that glutathione reductase is essential for neutrophil in order to do their job. Then once again in 2012 Ann et al. The same group they demonstrated that glutathione reductase is very essential for the formation of neutrophil extracellular traps. Then in 2019, Chenchameron et al. They showed that human polymorphonuclear neutrophil response to Burkholderia pseudomyeli in healthy and diabetic subjects. So, diabetic patients are very prone for the infection of Burkholderia pseudomyeli. The clinical condition is known as melioidosis. So, it is common in Southeast Asia. They are very prone for Burkholderia pseudomyeli. in the patient suffering from myelaidosis, what happens? The neutrophils of such patients, they are unable to phagocytosize the pathogens, and they are unable to undergo degranulation as well as net formation. Because of that reason, they will suffer. You can find sustainable infection by Burkholderia pseudomyeli in diabetic patients. Then, in addition to that, there is another clinical disease called chronic granulomatous disease, or CGD. It is a genetic disorder in which the NADPH oxidase is not working properly in the neutrophils of CGD patients. Neutrophils can phagocytosize the bacteria, but they are unable to digest the bacteria. So, because there is no oxidative burst in the neutrophils of CGD patients. So, because of that reason, we can expect malfunctioning of neutrophils both in CGD as well as myelaidosis. So, these are some photographs of myelaidosis clinical condition, especially in diabetic patients. The major problem with neutrophils of with myelaidosis patient, you can see decreased levels of GSH in the neutrophils. Neutrophils they cannot perform their basic function, especially phagocytosis and degranulation. So, because of that reason, they will have sustainable infection. So, in CGD, as I mentioned earlier, there is a defective NADPH oxidase enzyme. Neutrophil exhibits normal phagocytosis but fail to display microbicidal activity. So, because of that reason, CGD patients are more vulnerable for sustainable infection. So, all these facts, from all these facts, we can say neutrophils should be in a proper manner, or we can say, especially in the clinical conditions like CGD and myelaidosis, there is a dysfunctional neutrophils. If we restore their function, we can we can cure or we can curb the CGD problem as well as myelaidosis in diabetic patients. So, based on these facts, we can say a tightly regulated balance between RAS production and GSHG cycle in neutrophil is critical for an efficient battling against microbial infection. The use of molecules that can protect as well as enhance the antimicrobial competence of neutrophils during clinical condition is required, especially for myelaidosis and CGD. The usage of non-specific and low molecular weight antioxidant at high doses have failed in prevention or mitigation of several pathologies. It is already has been proved. But redox modulators targeting specific ROS mediated signaling pathways like melatonin are highly effective to restore the neutrophil function. So, based on these ideas, we selected a melatonin for our hypothesis. So, melatonin is a simple amino acid derivative, N-tamine, derivative of serotonin. It generally produces by pineal gland. In addition to that, we can find there will be a release of melatonin from other parts of the body also. The major function of melatonin, it maintains circadian rhythm, or it regulates sleep wake up cycle. Based on the literature survey, we can consider melatonin as a conditional antioxidant come redox modulator. And meantime, and also in addition to this, as I said earlier, we can find till now we can find so many GPX mimetics, but not GR mimetic. Okay. So, based on the literature survey, melatonin works at three different levels in a cell. First, at the level of plasma membrane, second at the level of cytosol, third at the level of nucleus. So, it works as a antioxidant as well as redox modulator. It is a direct activator of NRF2, and based on our results say that it can block glutathione of cellular component, and also melatonin is having receptors on every cell is having receptors for melatonin, MLT1 and MLT2. So there is a receptor desensitization, there is no toxicity by melatonin. So from all these facts we can say melatonin serves as a redox modulator. So we asked a question, can melatonin restore the defective neutrophil function? So for our hypothesis, we used these chemicals, melatonin, a conditional antioxidant cum redox modulator, and we have used 2-AAPA, a specific glutathione reductase inhibitor, a cell permeable specific GR inhibitor. It specifically inhibit the GR. Then we used LPS as a neutrophil activation inducer, and we used E. coli cell, GFP E. coli cell to study neutrophil phagocytosis, and we used antibodies against these two markers, CD63 and CD35. These are markers for degranulation. Then we in our study we have used PMA, 4-ball myristate acetate as a netosis inducer. So first we asked a question, can melatonin ameliorates H2O2 or hydrogen peroxide and or GR activity deficiency induced toxicity in human neutrophils? What we have done, initially we have taken human neutrophils treated with 2-AAPA, a specific GR inhibitor, cell permeable GR inhibitor, and we knock down the enzyme activity. So we can call such neutrophils as glutathione reductase activity deficient neutrophils. So we incubated neutrophils with 2-AAPA for 1 hour to make them as GR activity deficient neutrophils. Then we treated them with H2O2 for 30 minutes in order to induce oxidative stress, and we asked a question whether such neutrophils undergo apoptosis or reactive oxygen species get elevated or not. We found reactive oxygen species are get elevated in neutrophils, GR activity deficient neutrophils, when compared to H2O2 alone. When we treat with melatonin, we found restoration, and survival rate get increases, cell viability get increases in case of melatonin. From these data we can say melatonin can protect GR deficient activity deficient neutrophils under oxidative stress induced by H2O2 to a greater extent. So it also curb the apoptotic markers in GR activity deficient neutrophils treated with H2O2. Then we ask next question, can melatonin restores the neutrophil degranulation function upon GR activity inhibition? For this also what we have done, we treated the neutrophil with 2-APA and converted them as GR activity deficient neutrophils, and we challenged with LPS. So when we done that, the markers, for example, CD63 is a marker for primary granulocytes. CD35 is a marker for secretory granule. We have used these two CD markers as a degranulation marker for neutrophils. When we estimated CD63 and CD65, there was a decreased levels of expression of these two on the surface of neutrophils. That clearly suggests that 2-APA, when we inhibit GR by 2-APA, even though if we challenge with LPS, there is no degranulation, or we can say function of neutrophil get hampered. Same cells were treated with melatonin when significantly we found significant increase or restoration of CD63 and CD35 expression on the cell surface of GR deficient neutrophils. So this data clearly suggests melatonin it is restoring the neutrophil degranulation function in GR activity deficient neutrophils in presence or absence of LPS. Then we assess the next neutrophil function, that is pathogen phagocytosis, by counting the E. coli cells present in the media when we incubate GR deficient neutrophils with E. coli. And we found when neutrophils were treated with 2-APA, or we can say GR activity deficient neutrophils, they were unable to engulf the bacteria. So because of that reason, we found more number of E. coli cell in the media when compared to normal. When we treated with melatonin, the activity of neutrophil get restored, activity of function of neutrophils, that too in GR deficient neutrophils get restored back. So further, we have done some fluorimetric as well as FACS analysis in order to estimate the green fluorescent protein present in the E. coli cell and GFP in neutrophils which are engulfed the bacteria. So all these data clearly suggest that melatonin is restoring the neutrophil pathogen phagocytosis function in GR activity deficient neutrophils. Then we asked next question whether melatonin can restore the neutrophil function, that is netosis. So for this also, neutrophils initially treated with 2-APA, then challenged with LPS or PMA as a netosis inducer, the netosis levels are decreased, significantly decreases when compared to neutrophils alone. So in other words, you can say neutrophil formation in GR deficient, activity deficient neutrophils were low compared to normal neutrophils in presence of LPS as well as PMA. When we treat GR activity deficient neutrophils with melatonin and challenge with LPS and PMA, we found significant restoration of net formation in the GR deficient neutrophils. From these data, we can say melatonin is restoring all the three functions of neutrophils like degranulation, phagocytosis, and netosis. So we asked a question, what is the molecular mechanism behind it? So by oxidative burst is the major contributing factor for neutrophils in order to kill the bacteria, or microbicidal activity depends on oxidative burst. NOx, NADPH oxidase, is the enzyme major source for the production of superoxides. So we thought like since the ROS levels are getting decreased when you inhibit the GR activity, we asked a question whether NOx enzyme get inhibited by this event or not. So that's why what we have done, we treated the neutrophil with 2-APA, make them as GR activity deficient neutrophils, and estimated the NOx activity in them, and we found there will be a significant decrease in the NOx activity in a GR activity deficient neutrophils when compared to normal. When we add LPS, to challenge them, we found decreased levels of NOx activity in GR deficient neutrophils compared to normal neutrophils. Melatonin restores the activity of neutrophils. In addition to that, we assess the ratio between GSSG and GSH. When we inhibit the GR glutathione reductase, we found more amount of GSSG compared to GSH. More amount of GSSG, or we can say oxidized glutathione in the cell compared to GSH. So because of that reason, neutrophils, they were not performing, they were not displaying their normal function. Based on the earlier studies, we asked a question whether there is a based on the earlier literature survey, when there is a oxidized glutathione level get increases, cellular proteins, they undergo glutathionylation. So in order to prove is there any glutathionylation is happening to the cellular proteins or not, we did Western blot by using anti-GSH antibody, and we found there is a significant increase in the glutathionylation of cellular proteins. So then there are two events are more critical for neutrophil function. One is NOx activity, another one is actin. When cell undergo degranulation, actin dynamics will vary. When cell undergo phagocytosis, actin dynamics get changed. When cell undergo netosis, there also actin dynamics will vary. So we asked the question whether these two proteins are undergoing glutathionylation, thereby their functions are modulating. So for that we used immunoprecipitation assay, and we confirmed that there is a significant increase in the glutathionylation of actin and as well as NOx enzyme, particularly P67 FOX. In GR activity deficient neutrophils, even though we challenged with LPS. In presence of melatonin, the glutathione levels are significantly get decreased. This clearly suggests melatonin, by blocking glutathionylation of actin as well as glutathionylation of NADPH oxidase, it is restoring the neutrophil function. Due to time confinement, I didn't show. I'm not showing some more experiments. Like in addition to that, we have shown that glutathione directly activate NRF2 transcription factor. Thereby, other antioxidant enzyme levels are get increases in presence of melatonin. So we did some animal experiment. I'm not showing much of the data. In animals also, there are 6 different groups. First group is saline treated, second group is 2-APA treated, third group is LPS challenged, fourth group is 2-APA plus LPS, fifth group is 2-APA plus LPS and challenged with melatonin. The last group is melatonin alone, and we assessed total RAS in neutrophils isolated from the experimental animals and plasma DNA for net estimation, and cytokine fluorescence we assessed. All these experiments in all these experiments, melatonin restores the levels of DCFDA. So from all this data, we can conclude that when we can conclude that GR play an important role in regulation of neutrophil function, especially degranulation, phagocytosis, and netosis. Melatonin effectively restores ROS generation, phagocytosis, net formation in GSH as well as GR activity deficient neutrophils. In one set of experiment we blocked the GR. In another experiment we blocked synthesis of GSH by using B.H.U, a specific inhibitor for glutathione synthetase. Thereby we make neutrophil to have very low amount of GSH. In both the cases, melatonin effectively restores the ROS generation, phagocytosis, and net formation. In this study, we have showed an uncharted property of melatonin that modulates the redox status and aid to execute neutrophil function under cellular GSH depletion and GR activity deficiency. Melatonin could be an appropriate candidate for auxiliary therapy to treat microbial infection, including viral infection even under chronic disease condition. So once we publish this data in the JPR, after that we found there were 33 review articles on melatonin, usage of melatonin in COVID-19. Some group tried melatonin in clinical trials also to know its efficacy against COVID-19. So initially we included a lot of information about COVID-19 and melatonin in our research manuscript, but journal editor he was not allowing us to keep those things in our article. So this is the publication what we made based on the results what we get. It got published in June 2020. We submitted in April 2020, that too in first lockdown, COVID-19 lockdown. And I take this opportunity to thank my mentor, Professor K. Kemparaju sir, University of Mysore. He is my PhD mentor and well-wisher. Still, we are having a lot of association with research. And this is the guy, Dr. S.K. Naveen Kumar. He only did all these experiments with the help of Dr. M. Shekhar, Dr. Jagdish, Manikanta, and Vishalakshi. We got funds from DST, SERB, Institute of Excellence, University of Mysore. Thank you for patient hearing. I'm happy to answer for your question. Thank you once again.
Thank you, sir. It was a very elaborate talk on different mechanism, right from the beginning till the culmination of different type of mechanisms. And I very thankful to you that you have joined us and enlightened all of our participants. Previously, Dr. Kemparaju sir's lecture was also people were able to hear, and he also elaborated the things very in a very simplistic manner, just like yours. So, and I request all the participants to ask their questions as the session is open for all of them for discussion. Thank you.
Very good morning, sir.
Please introduce yourself to the speaker and then.
Yes, sir. So, a very good morning to all, and this is Mahesh from University of Hyderabad. And your talk is very informative and nice talk, sir. And I have some couple of questions, and just basic questions. So, in some context, every cell can act as a immune activity. I mean, immune functional. But what is the typical function of the immune cell deviating from the normal cell? That just I want to know from you, sir.
As I mentioned earlier, there are cells which are involved in innate immune response as well as adaptive immune response, whereas basic cell cannot do such jobs. For example, if you take WBC and platelet as an example and fibroblast as an example, fibroblast is non-immune cell, whereas platelet and neutrophil as a immune cell. What platelet does, it cannot kill the microbe, but it bundles up the microbe and it gives that thing to WBC. WBC can engulf the entire bacteria and it will digest, first thing. Then if you take WBC as an example, WBC itself is a phagocytic cell, it can engulf. And in RBC, in case of fibroblast, it is not having oxidative burst, or we can say it is not having that much of reactive oxygen species producing capacity. There's a huge change between immune cells and basic cells. For example, NOx activity when compared to neutrophil, we can find very less amount of NOx activity in fibroblast. First point.
Yes.
And these cells cannot undergo degranulation. They don't have granular thing.
Okay. Yes.
And they cannot phagocytosis. They cannot phagocytosize anything.
Yes.
They can
Okay.
exchange something between the cells, but they cannot grab.
Okay. And why I got this doubt is, I mean, every cell can release like, inflammatory mediators, like, pro-inflammatory, anti-inflammatory kind of. So those cells we nowadays are considered as the immune cells also, no? So, I mean, in future, any evolution of the new cells kind of, they have the immune functions.
I
So,
don't know. I don't know exactly. I don't know. But T cells are known to produce a
yes.
lot of pro-inflammatory as well as anti-inflammatory cytokines. Fibroblast is they are unable to secrete.
Yes, and coming to your melatonin study, sir, I think it is looks very interesting thing. And coming to the animal experiments, and what is the basal level of the melatonin levels in that animals? So that and moreover, did you found any abnormalities in after treating with melatonin and behavioral changes and all?
No, no, no. Because, as I said earlier, melatonin is a very good molecule because melatonin
Yes.
can easily enter into the cell, first point, or it
Okay.
can enter the cell through receptor, specific receptor, MLT1 and MLT2. These are the two specific receptor for melatonin. And also there is
Okay.
a receptor desensitization concept. When cell says, I am done with the melatonin, I cannot take more, even though there will be elevated levels of melatonin, cell cannot take because receptor is get desensitized.
Okay.
So based on that, melatonin cannot induce. Generally, people will think that if you take more melatonin, it induces sleep. No,
Yeah. Yes. Yes. Yeah.
first point. And also
Yeah.
there were so many animal experiments in that they infused more than to say 2-3 grams of melatonin to the animals. They didn't find any sort of changes in them, behavioral as well as pathological changes. They didn't observe.
Okay. The last question, sir. And your experiment, I think you have treated with the system with LPS. I
Yeah,
think so.
yeah,
So, as of my knowledge, I think LPS can activate the TLR system, innate immune response.
yes, definitely.
So, I mean, have you found that any TLR changes other than this melatonin activities in that neutrophil activity?
We didn't change that, but
Okay.
we evaluated the functions of neutrophils.
Okay. In one
Evaluated,
direction.
yeah,
Yeah.
in one direction. We have evaluated. What we have done, we made neutrophils as a GR activity deficient neutrophils. Then we
Okay.
challenged with LPS,
Okay. Okay.
and we didn't find any sort of when we challenge it with LPS, there will be a degranulation. There should be a degranulation.
Okay.
We didn't find.
Okay. Thank you so much, sir. Thank
Thank
you.
you,
Very
thank you,
nice talk, sir. Yes.
thank you.
Good morning, Professor. This is Dr. Krishnaveni from Mananmani Sundarnar University.
Yes, ma'am.
Very nice. I started from basic and then you entered into the small query. Like you said, about netosis. So actually it
Yes,
is a protective behavior by the neutrophil. But in lot of literature I have seen that most of the pathogenic organisms, they use this netosis as their, that is, take it as an advantage for their disease progression.
It depends on
Isn't it?
it depends on pathological conditions, ma'am. Based on that, we can say when there is a what to say when any cell yeah, I am talking about only netosis, neutrophil thing, but other cells are also undergoing. They are ejecting their DNA. That process is known as etosis. For example, endothelial cells, they release their DNA. Like that, fibroblast under necrosis, they can release their DNA. Once they release their DNA, there is no DNA's activity is very low in our circulation. DNA's activity is very low in circulation, very low in circulation. So system cannot eliminate or digest the DNA. It can digest the DNA to some extent, but not much. So macrophages only can grab the DNA, and macrophage is having DNase enzyme, it can digest it. So in such cases, what happens when DNA get accumulated in the
Okay,
circulation,
thank you, sir.
that leads to many complications, man. Whereas this one, how to say, whereas in case of neutrophil, netosis is a protective function. So in order to kill the microbe, it has to undergo netosis. If neutrophils is failed to undergo netosis, it cannot kill the pathogens in an efficient manner. Whereas in case of resident cell, if they undergo netosis, or we can say endothelial cells, if they undergo netosis, that is a detrimental factor.
So in that case, is there anything which can modulate this? It has to it has to stop after some time or something like that?
I think we cannot say like that, but there are two types of netosis. One is suicidal netosis, another one is vital netosis. Vital netosis happens within 90 minutes. So if neutrophil will eject its DNA within 90 minutes, but neutrophil won't die. Whereas in suicidal netosis, neutrophil die by ejecting its DNA. It takes around 3 hours. So in case of higher animals, there is no vital netosis. Only suicidal netosis is there in higher animals, except in snake venom. So there is one snake venom toxin from Indian cobra. That toxin only induces vital netosis in higher animals. Whereas in microbes, there are few examples which can induce vital netosis. So that's why as a as a biochemist, we can say wear and tear is good. For example, I don't know whether I have to discuss with this matter or not. For example, females, we cannot find, females are very they are not prone for cardiovascular disorders when compared to men's, as my knowledge. When after menopause, they are prone for cardiovascular disorders. Before menopause, they are not prone for cardiovascular disorders. Whereas men's, irrespective of age, they are prone for cardiovascular disorders. It is because of net formation, nets in the circulation. Because every month, as a menstrual cycle, they will discharge lot of blood components outside the body. Whereas men's, they don't have such event. So that's why while shaving, if they get cut, it is good for, according to me, so that it can activate macrophage DNAs. So that thereby hello? Hello?
Sir, you are audible.
Yes, sir. Yes, sir. Yes, sir.
Yeah. और उसको बड़ा कर दीजिए. Sir, Azim Ali from Azim Ali wanted to ask that I want to know during chronic disease neutrophil recruitment how ameliorates the disease condition.
Sir, can you repeat once again, sir, please?
Yes, sir. Yes, sir. He wanted to know that during the chronic diseases, how neutrophil recruitment ameliorates the disease condition.
Sir, we can say in COVID condition, there will be a dysfunctioning of neutrophils. So if you restore the functioning of neutrophils, we can decrease the disease severity. So one such thing is melatonin. We can give melatonin as an auxiliary therapy. There is a professor by name Russell J. Reiter. He is the founder, editor of Journal of Pineal Research. He is in Texas. During COVID-19 situation and all, he educated the entire city with his knowledge, and he circulated, or he has given he gave melatonin tablet to every individual in a city, and he educated the things, and he found very good results from melatonin.
Sir, the second question is from Priyanka.
Yes.
That she wanted that if you can elaborate a single disease which is a result of overreaction of the immune system.
Sir, overreaction of immune system, we can take any autoimmune disorder, sir. For example, rheumatoid arthritis. It is a very big thing. Yeah, and there also there will be a lot of nets formation.
Yes.
And the best marker for rheumatoid arthritis is citrin-related proteins, which are present in the histones. When cells release the DNA along with histones, then you can find citrin-related. That is the best example for overreaction of immune system with respect to net formation. I hope I answer our question.
Yes.
I don't.
Sir, Chetna Jamra wanted to ask that is there any vaccine developed against
Melioidosis. No vaccine, sir. No vaccine. We try to get the clinical samples to know by using neutrophils from patient suffering from melioidosis, and if we provide melatonin, can it restore the function of such neutrophils? We wanted to know that effect, but due to COVID situation, we could not able to get the samples, because in Karnataka only Manipal hospital is having some samples. Some patients, some patients, they are visiting to the Manipal hospital. In a month we can get 2 or 3. We are trying to collaborate with them. In future I may get some blood samples from them, then we are interested to evaluate that such things.
Hello, sir. This is Iban Lahdem from A.M.S. Kalyani. Sir, my question is exactly how does melatonin helps in this? Does it like affect the inhibitor, remove the inhibition? How does it really, works in ameliorating the the inhibition by this AAPA?
AAPA. Okay, madam. Once if you, I will take neutrophil as an example. When you treat neutrophil with 2-AAPA, it blocks the glutathione reductase enzyme. When you block the glutathione reductase enzyme, there is no conversion of oxidized glutathione to reduced glutathione. In a cell, GSSG or oxidized glutathione level get increases. When levels of oxidized glutathione get increases, it will non-enzymatically glutathionate the cellular proteins. For example, actin. If once actin get glutathionated, cell cannot use its actin dynamics to move. For example, cell cannot degranulate, cell cannot phagocytosis, cell cannot eject its DNA, because actin dynamics are get shut down. That is one thing. And also, if NOx, NADPH oxidase enzyme get glutathionated, its activity get modulated. In other words, you can say the NOx enzyme activity goes down. When NOx enzyme activity goes down, superoxide level generation goes down. When there is no superoxide generation, microbicidal activity of neutrophil goes down or won't work. So this is the reason. How to say? First thing, it quenches the RAS, okay, as an antioxidant. Second thing, it blocks the glutathionation of important cellular proteins like actin and NADPH oxidase enzyme. Third one, it works at nuclear level by activating NRF2. It boosts up the antioxidant system or machinery of a cell. Thereby, it protects the cell from oxidative stress.
Okay, thank you, sir. And this inhibitor is like a permanent like inhibitor of the GR, or
Which
is
one?
it like
2-APA?
yes. Is it like a permanent inhibitor or
99%, 98% it inhibits the GR, glutathione reductase. In addition to that, we can there are off targets. Thyroidoxine reductase also get inhibited by 2-APA to some extent.
okay. Thank you, sir.
Thyroidoxine reductase. Thank you, ma'am.
Any more questions?
Okay. Yeah. Sir, Vinaya Kruti asked that can bats' immune response to viral infection studied to discover some targets of drug development so that it could help fight with like SARS-CoV like infection?
Oh, maybe yes, sir, but I don't know. There is a huge difference between bat and human system. We don't know how exactly it works with us. We don't know. I don't know. Sorry, I don't know. I don't know much about this, sir. Sorry.
Okay. So I think this area is still open for our participants to explore, as many of them are budding scientists, so they can definitely explore in some area,
Yes.
and we'll definitely get some inspiration from you, sir, so that they can start their research and make it to the heights that looking you as an example.
Thank you, sir. Thanks a lot, sir. Sir, namaskar, sir.
Oh, no, sir.
Yeah, thank you very much, sir. Dash sir is the inspiration for me because when I came from US, I started thinking of platelet biology. I didn't know about Dash sir up to 2012. Once I read his paper related to platelet biology, I became a big fan of his research. Till now, I am keep on reading his paper. He is a source of inspiration for me to continue in platelet biology. Sir is doing excellent research. It is a if we follow his footsteps, that is more than enough. We can definitely we can come up in our life. I would say follow Lasser and read more number of papers and especially read papers from his group. They are unique in nature, so that you will come to know how exactly design an experiment, where are the gaps are available within the research idea. So if you start filling those gaps, you can get a nice publications as well as you can learn many things.
Thank
All the best.
Dr. Girish.
Sir, please, sir.
If I have inspired you, now it is your turn to inspire this audience. You are really the inspiration for them. I was just enjoying your talk and your interaction with the audience. And basically, I am addressing to this audience that if there are 3-4 groups really aggressively working in this area, apart from our group, here is Dr. Girish. Yesterday, you have listened to Professor Kemparaju, wonderful orator, wonderful teacher, and he's a teacher or mentor of Girish as well.
Yes, sir. Yes, sir.
That group in Mysore, Girish
Yes,
group in Tumkaru, which is near Bangalore, our group. And yesterday also, people have listened to Professor Guchai from RCB.
Yes, sir.
And Jahid Asraf from
Yes,
Jamia.
sir. Yeah.
So
Yes, sir.
these are the 5 groups, and it is so much limited group of people who are really investigating in this area.
Yes, sir.
So the purpose of this workshop and overall our goal is that we should inspire more of the budding scientists who aspire to do something more to venture into this area.
Yes, sir. Yes,
So
sir.
here you have a lot of responsibility to bear on. So
Thank you, sir.
sir.
Thank you, sir.
See you at the other end again.
Yes, sir. Definitely, sir.
So maybe
Thank you,
next
sir.
physically meeting at
Yes, sir.
B.H.U.
Definitely, sir. Definitely, sir. Thank you, sir.
Okay.
Thank you.
Thank
Thank you, sir. Thank
Thank
you.
you, sir. Thank you, sir.
Thank you.
If there is any more question, please please ask. You can also type your questions in the chat box. So, sir, for last 3 days, all our participants were listening only to the platelet signaling and different techniques where we can go for, but some of us, some of them really wanted to know about crosstalk between platelets and other cells in the blood, including neutrophils. So I hope that their queries and their quench for knowledge in this area definitely would have been uh, 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 with your talk with on neutrophils, because many of them asked us that whenever there is inflammation or infection, then how this crosstalk will happen. So
Yes.
your talk elaborated in different pathways where different blood cells will have a crosstalk. So I think that was a very wonderful talk from your side,
Thank
and
you, sir.
I thank you again.
Thanks a lot, sir.
You may please continue in this session. Our next speaker is Dr. Hina from
Yes, sir.
ICMR. So
Yes,
she'll
sir.
be joining us soon.
Yes, sir.
Yes. Thank you, sir.
Please, sir. Thank you, sir. Thanks a lot, sir. Thank you, sir.
Okay.
Thank you. Thank you.
She's there. Sure, sir. She's having some network issue. Yes. So if the participants have question from the previous section, they are all welcome to us. We have all the experts here right now. If you have any questions regarding the technical part, then also you can ask from intermetal microscopy, from flow cytometry, or any other. Please feel free to ask.
Am I audible?
Yes. Okay. Let me just,
Good morning, sir.
Hello, Hinaji. Good morning. Namaskar. So, to the audience, just I want to speak that for this this workshop is for the to spread the awareness about platelet and thrombosis research. And this is an idea-driven workshop. It is not a gadget-driven that you are to know the mechanism of operation of a principle of equipment. It is just a concept, overall concept of how one can carry out research in the area of platelets and thrombosis biology. So when it is an idea-driven, there are two types of research. If it is a fund-driven, idea comes later. पहले पैसा ले लो, बाद में idea मिलेगा. Idea-driven research.
Mute everybody. Mute everybody. Mute all. नहीं अभी भी. It's not ours. Please, I request all the to please mute yourself.
Please mute everybody. Yeah. So what I'm speaking that for carrying out a good research on a good idea, if the idea should come fast, and finance is required. But I see that at many places people go and I visit them, they told that we don't have a fund, etc. How can I carry out research? If you have a good idea, you can start from scratch. All of us have started from scratch, and gradually you collaborate, cooperate, get grant from your organization, little bit tidbits. Even I have invested my own money at the prime of my career also. So anyway, you have to build up a career, then research will fund will come. Now, being associated with many funding agencies over a period of years, I have known very well that it is a very difficult task to allocate funds to eligible persons because there is a huge competition. In a limited time, you are to judge many proposals. All the proposals are quite good. There is a financial constraint. So, in the other side of the table, that the other stakeholder is a funding agency who can fund our research, but they have a real difficult, very hard task, and they have to do it very transparently as well. So with this backdrop, I thought that you should listen to a person, a scientist who is engaged in the funding, work in the funding agency, and I am very happy that today we are having Hinaji here. I think it must be a very busy schedule at this 11:30. I know she is not available, used not to be available in her chamber whenever I visit. She is in this meeting or that meeting. So luckily we have captured her today. And so I would say that I have seen a generation of scientists in ICMR, and this young generation of scientists whom Hinaji represent, young and dynamic and very focused and to the point. So I see that in these people, new scientists, new Gen Next scientists, there is a great future for the funding agencies as well. So with this introduction, I would request Dr. Kamlesh to introduce Hinaji before she starts her talk.
Thank you, sir. Now I will introduce our next speaker, Dr. Hina Tabassum. She is currently working as a scientist and program officer in Basic Medical Sciences Division of ICMR headquarters, New Delhi. Dr. Tabassum completed her PhD from University College of Medical Sciences in 2008 and completed her postdoc tenure at Germany between 2008 and 10. She is a DBT BioCare awardee and recipient of DST Cognitive Science Initiative and UGC Postdoctoral Fellowship. Her research interests included development of preclinical models of brain injury. She has more than 45 publications with an H-index of 16 and more than 750 citations. Dr. Tabassum has involved in PI, co-PI several as a PI and co-PI in several external research projects. Recently, she was a part of ICMR Secretariat that disseminated ICMR guidelines for GCP, that good clinical laboratory practices 2021. She has initiated National Hospital-based registry on venous thromboembolic disorders at ICMR. She has been coordinating the external research in the field of nanomedicine, anatomy, hematology, toxicology, traditional medicine, and physiology disciplines at ICMR. So whatever our participants are aiming right now, right from nanomedicine to hematology and many of the thrombotic research, definitely she is the one who will be screening our research. So definitely she can guide us on how to write the research proposal, what at all we will be needing to do so that our research proposal should look lucrative and many of our budding scientists who are part of our participants, so they will definitely be benefited and will be and her help will able to make their careers more flourishing. Thank you, ma'am. With these words, I welcome Dr. Hina for her elaboratory talk on funding and other aspects. Thank you, ma'am.
Thank you, sir. Good morning, Professor Das, Dr. Kamlesh, for inviting me to this event, and heartiest congratulations on the successful event to you all. Good morning to everyone who is listening to us right now. So, from the kind introduction that Dr. Kamlesh gave on my behalf, I was into active research till 5 years ago. After that, I am more into science management and administration. And as Professor Dash introduced, I will be giving a brief talk on what kind of funding opportunities Indian Council of Medical Research provides to researchers at all levels, pan India. And also, we have our new DG, Professor Rajiv Behel, who has come from WHO, and he has recently joined as Secretary DHR and DG ICMR. So, what he is having in his mind regarding the research that is currently going on in India and how to upgrade it in terms of funding. So, I'll be giving a brief on that as well. So, not taking much time, I'll share my PPT with you.
Okay, ma'am. The screen is visible, ma'am.
Is it visible?
Yes, ma'am. Now it's visible. Make it into the full screen mode.
Yes. So, and directly coming on to what kind of research grant schemes that ICMR provides to the interested and eligible researchers. We have two kinds of funding programs. One is intramural research, as most of you would be aware that ICMR, apart from funding the universities, institutes at national level, also host 28 intramural or its own institutes, which are based on certain diseases, certain themes pan India. For them, we have a separate wing which provides intramural research grants to those institutes exclusively. And I will be more emphasizing on the extramural research part, where all eligible Indian citizens are, they may apply. So, first one is, I think the most of the audience belongs to this section of our researchers, that is students. So students can be divided into people who are pursuing their masters in science and they would like to do PhD, or they are already pursuing PhD, and followed by a postdoctoral or a research associateship in their career. And apart from that, we also have a separate schemes that are dedicated to medical students and professionals. So when I talk about fellowships, so we have something called as junior research fellowships. That it is an exam based scheme where people, just like you have JRF for CSIR, so we have ICMR JRS, and I've also included links where you can read, or you can directly go to ICMR's main website and you will easily find all these under the title grants. So we have JRS, which is provided to all the eligible candidates who have completed their masters in sciences, and they may apply and sit for the exam, and if they clear it, they are eligible to get this fellowship. Apart from that, we have independent senior research fellowship, which is provided to someone who has already done their masters and they are working in or continuing in a lab for at least 2 years, and they have at least one publication. They are eligible to apply for this. Uh, how the scheme works, we have an online portal which opens once in a year, which where a person can apply for this kind of fellowship. And for this, you need to have a host lab and a proposal, an independent proposal, which will be evaluated by the peer group, and if found suitable, you will be funded for at least 3 years, which may be extended on your request and based on your performance in last 3 years. We also have research associateships. This is provided to people who have already completed their PhD in sciences, and the process is same. Once the portal is open, you may apply for this particular grant scheme, and again, you need to have a proposal from along with a host institute or a host scientist or principal investigator with you, and this is also given for at least 3 years. We also have a postdoctoral research funding. This is particular for people who have also done their PhDs, but this particular funding will only be allowed at ICMR institute. So if this is just like a research associate, but it is only given at any of the 28 institutes that we have pan India. You can apply, you can find a host there and apply for this. And in this, usually a proposal is based on the work which was already done in your PhD program. So this is one condition with this particular research funding if you opt for it. And as I said, we have specifically dedicated funding schemes for the medical students and professionals. So we have schemes for MD, MS, PhD programs. We give support for thesis writing for MD, DM, DNB students or MDS students. So they can actually go and check the websites for the eligibility criteria. We also recently started with nurturing clinical scientist scheme. So this is particularly for fresh MBBS or BDS students who have completed not more than 2 years of their degrees, and if they are interested in doing fundamental, basic or clinical research, they may apply for the same. We also have every year short-term studentship program, that is STS, and this is again particularly given to MBBS or BDS students anywhere between their first to fourth year before they are done with their fourth year final exams. So they may also check if anyone is interested in that. We this scheme, the last scheme under which is particularly given to the medical professional, is something called as medical innovation scheme. This is again a very restricted option for anyone who does not work at ICMR, because this is given to in particular scientists working at its own institutes. Now coming on to the major emphasis which is funding a lot of researchers in India, is extramural research, where different types of proposals are submitted to ICMR every year, and people look forward to it. And this also opens along with the SRFs and RA every year, once a year in December. So hopefully we see this cycle soon. So we have something called as ad hoc individual investigator oriented project. Here a PI who is with PI I mean a principal investigator or a faculty who is a permanent member of an organization or an institute or university. They are eligible to apply with the condition that you should have at least 3 years. It is open to everyone till they have at least 3 years before they superannuate or retire from the organization. So for ad hoc projects, the funding limit for now is up to 1.5 crores. So these are more. When I talk about SRFs, RAs, postdoctoral research scheme, or the ad hoc projects, so these are usually PI oriented or the fellow oriented. It means the idea comes from you, and we have a review of that proposal by the experts, the subject experts in the area, and they find it whether suitable or not for funding. So the idea comes from you, but we have something called as, this is specifically for the faculties here. So we something called as task force projects. The task force project is based on the fact that we want to solve a predefined problem. So it is centrally identified. It means we have one common problem and we have multiple centers working along with it. So this is a long-term project. It can go on for 5 years if recommended, and everyone who is part of it, they work on a common goal with certain objectives that will be predefined at ICMR. So we also have provision of formation of a registry or a database for a problem. So that can be hosted at ICMR in public domain, which will be available for other researchers for their information in future. There is also a provision for something called a Center for Advanced Research. Now, Center for Advanced Research is basically for capacity building and to support sustain high quality research. In general layman terms, it is an opportunity which is given to researchers all throughout India, where they it can be from ICMR that we float a call that we need somebody to work on. Let's say SIR has one Center for Advanced Research on thrombosis. So we would like to have a detailed research, both fundamental and clinical or otherwise. So he has taken the responsibility for establishing this center at B.H.U, where he proposes to have several studies under this particular special center, and based we expect on the success of this center that after 5 years B.H.U will be able to adopt it and continue it further in future, in particular focus on thrombosis. So this is how Center for Advanced Research may also be applied for, and can be established at any of the eligible institute organization with but for this is usually given to a PI who have shown lot of credibility in that particular subject area. So all of these proposals, they go under a common review mechanism which has the program officers who are involved in that particular subject, they, as per their expertise, they also screen those projects. Then it goes through external peer review. We have something called as project review committees or project review groups or expert committees or technically advisory groups. So these are group of people that have been selected by ICMR to look into the applications for funding that we receive here. So we go through each and every application that has been received for administrative completeness, followed by the technical or the scientific soundness. And it is done for even for the fellowships, and a little more stronger for the ad hoc proposals, the task force project, and the Center for Advanced Research. We also have international collaborations. So these come again. This is for the faculties who are permanent positions or permanent jobs at institutes and organizations. So we time to time give out international collaborative calls. We have MOUs with European countries, with US, with Japan. Recently we had a MOU with Korea. So these collaborative call can be divided into that, maybe a workshop or a training program in a subject area may be given out, or recently in the subject of toxicology that I coordinate at ICMR, we had floated a joint call between India and DFG, that is funding agency for Germany, submission of proposal in the subject area of toxicology. So similarly, you can check on the website time to time. We have these kind of international joint calls with the international funding agencies. We also have opportunities for seniors. For seniors, I mean people who are veterans, people who have some amount and years of expertise in for a subject area. So we have something called as adjunct faculty scheme. It is both ways. A scientist from ICMR or vice versa, scientist or a faculty or a professor or a medical professional from non-ICMR organization can be hired to be an adjunct faculty to support. If somebody from non-ICMR organization is coming to us, so they will be helping the researchers at institute which has appointed them as adjunct faculty to them in their research work. And similarly, if somebody goes out from here, they will be doing the same at a non-ICMR organization. We also give opportunity to people who have superannuated from their position to become ICMR emeritus scientists. So you can check out. I have already shared my PPT with links, and otherwise you can go to the main website for ICMR, icmr.nic.in, and you can check that what are the conditions for applying to this particular scheme. We also have for eminent scientists from biomedical field or life sciences or medical field per se to become ICMR chairs. So the chairs are we have two chairs, Dr. C.G. Pandit National Chair and Dr. A.S. Pentel Distinguished Scientist Chair. This is in particularly reserved for people who have shown tremendous credentials in their field in Indian science, and they are appointed from anywhere from 3 years to 5 years, and they are part of ICMR family, and their knowledge can be taken up by any of the researcher or the scientists who are working with ICMR, and they are also part of various guidelines that ICMR tends to prepare as and when we talk about research and science. We also have opportunity to have clinical training and translational research workshops. So it means, right, we also give support, financial support to people who want to organize a training or a workshop where you can have a hands-on training for any clinical procedure to medical students, or a translational research workshop in context where maybe a vaccine lab or a lab whose focus is to develop an intervention for a medical condition per se. So that may have a positive translational value to the society. So if you have an idea based on that, you may apply for this kind of funding to us. Unfortunately, post-COVID era, we have stopped giving funds for conferences, seminars, symposia, CMEs, or any short course funding that was available before 2019. We also support students and faculties in for having the travel in like international travel by non-ICMR scientists, which is currently suspended along with the conference grant. But ICMR and DHR, Department of Health Research, does have collaborative scheme where Indian biomedical scientists, depending on their age, below 45 or the senior people who are above 45, they may apply for a short-term or a long-term international fellowship if they have a host institute abroad. So it can vary from 2 weeks to 1 year. So this was a short note on what kind of funding or grant research scheme, whether it is a full-fledged fellowship or a workshop organization or a travel grant, depending on your PhD or a MBBS or MD qualification, what are the options available to you? I would say writing a grant is also a key factor in your success to get this grant. So you have to play by rule. It means we or any other funding organization throughout the world, if we talk, we have the guidelines. So before you apply to us, please get those guidelines, read it properly and follow them, because this is also one of the biggest reason or cause of failure of an application that we have been receiving in last 2 years. People don't read the guidelines. They write whatever they want. They do not follow what kind of word limit they should follow. What the question is being asked when you talk about writing a proposal. If somebody is asking you a translational value, it should be as crisp as 2 lines or 3 lines. It doesn't have to be a story of 1 page. And as I said, I would like to spread this desire of DG ICMR that now Government of India is also focusing on one factor, that although fundamental research is important, but now it's high time that whatever we do, it should have a short-term outcome. It means what will come out of it after 3 years. But what will happen to it after 3 years. So you need to come up with your answer if somebody is going to apply. When the new portal opens, you will find this question: where do you see this study or the results of this study after 3 years when you're done? So it won't be accepted if you say, I will be done with my thesis, I will be done with my 3-year project, that's it. No. So, telling that high impact publication will do, but what about who will be the buyer of your study or your research? So you need to focus on the future of your work. Why are you doing it? What is the need? And don't write that because it's not available in the literature. That would be a very poor explanation of you writing a proposal, submitting to ICMR with the hope that it will be funding because it is just novel. Not every novel thing has to be worked upon. It's not necessary. If you're working on something existing also, it is fine. But why are you doing it? What is the need of doing it? And what do you expect when you're done with your even a small proposal like a SRF proposal or a STS proposal? Your study, how it will benefit the scientific society after, once you're done with it. So just a publication will not help. So be prepared. This year the portal will have guidelines and the format will have major revisions to it. So hopefully the guidelines will be able to help you out that who can apply and what needs to be done at your end. So you have to be very prepared. So this is also important for students. Faculties will have a hard time definitely, but even SRS and RA who are hoping to apply this year, they should keep in mind that we will be coming up with these questions, and unless you have satisfactory reply to these, they will not be screened in for the review or funding. So this is just a gist of how we process the grant. So we have a grant application to our portal every year, or there are specific calls for faculties time to time, which are based on any diseased condition or any medical condition or need of the hour. So they are, but everything is available online. Once we have your application, we check for administrative completeness. If it is all right, then it goes under review to expert committee. This year, we will have more than one review of the proposal, and finally, either you have good news or otherwise. So, and one more thing, people, apart from the guidelines, you need to focus on deadlines. So people start submissions at 5 o'clock on the last day, and they expect us to keep the portal open after 5 o'clock, and then they say कि मेरा application जमा नहीं हुआ. So you have to have both the things ready, the guidelines and the deadlines. But this year it is going to be very tough because we have an international representation at DGship, and he has come with the notion that why not our research can be upgraded to an international level where our postdocs, our medicos, they go outside India and they perform so well. Why don't they perform same in our own country? So we will handhold, we will help you in improvisation if your project is selected. But from this year, it will be very difficult to have a selection, whether it is a SRF or a CAR project. So I have just given a small brief on what is going to happen to the portal this year. There will be lot of changes, and we really expect you to abide by them if you really want us to at least review the application. So they will not be reviewed that whatever comes and we review. So if you have not submitted as per the instructions provided to you, it will not go for the screening. In the screening, a group of experts will check for the scientific soundness as we expect. And if it goes through them and they are okay with that, then it will go to a next group, the higher group, the expert committee on the subject area for the final recommendation. So this year we are thinking of having a tiered screening instead of only one. So accordingly, we will put up the guidelines for everyone. But Government of India is now focused. It is an now government issue that we cannot be funding anything that comes to us, whether it is as simple as basic research. If you're talking about basic or fundamental research, then also you will have to come out with original thought. So when I talk about expected outcome or sustainability of your results after 3 years of your study, it doesn't mean you give us some scientific gibberish that it will do that or this. As a layman, if I ask you, whatever you are planning to do, whatever you are proposing to do, it should have an impact. And how will it impact me? If your mother asks me, क्या करते हो, क्या study करते हो, इसका क्या निकलेगा? So you give us your viewpoint in those 2 points. What is the expected outcome of your work? And it should not be high impact, because we will be raising the bar for PIs at least. So it will be very tough for the faculty that and similarly for RAs, you will need to have a high impact publication before your application is eligible to undergo even screening. So that impact number will be still it's under process, that what should be the good number, what should be the impact factor of at least one publication that needs to be under essential qualification for someone to apply to us. So these will be some screening points at the very first step of your application. So these are the key points when the expert committee evaluate a research proposal. The significance, that what question you're addressing, what is the importance of it, and how it will help in advancement of the current knowledge that already exists in the system. So that hence you cannot actually just plainly say, because there is no supporting evidence of using this molecule, this plant, or this technology in literature, hence I am doing it. So it's a big no. So you have to answer this question with lot of thought behind it, that why are you doing this research? And this is not only for faculties, it is also for SRFs and REs. So when we talk about an ad hoc project, a task force project, or something with higher funding to attach to it, then we also look for qualification of the investigators. When we look into SRS and RS, we would look into the profile of their host or their guides. So again, innovation. So it doesn't, when we say novel, it doesn't mean it doesn't exist in the system. So most of the, at least the fellowships, they are based on the thought, whatever I am doing, it is just absolutely new, de novo. It doesn't. They biasly chose out the references, the bibliography, which is actually similar to the work they are doing. And this has been checked by the expert committee this year. That is why the number has gone down in terms of success of an application which were funded. So you have to have this in mind. You can do something which is being done, but why do you want to carry that out, and what will it provide to the science, to the society, and the people who are going to follow your research? So this has to be kept in mind when you apply to us. So, and your approach means, is the idea or the proposal actually feasible with the infrastructure, with the credential of your guide or the PI itself? What is your strength? Let's say your strength is in, as sir is working on, or the theme of this platelet science or thrombosis, but you want to work on something else. So how would you justify? So you want to work with maybe a plant chemistry. So you might be a scientist, but you're not an expert in the area. So you have to keep in mind, for just for the sake of novelty, you cannot just start something new. If you have a novel component, it should have an association with your experience or expertise. Ethical approvals are also a big problem. People tend to wait for ethical approvals. Once everything is done, then they start applying. It should be other way around. You should start your ethical applications procedure at your institutes the moment you apply with us, because it takes a lot of time. I understand some of the institutes might not give ethical approval on a project which is not recommended or so, but at least the organization, institutes, or university should have some kind of mechanism that you can at least keep it on hold the moment it is okay for the funding. You may give a go ahead, but I think if a researcher or a PI has some kind of possibility of carrying out a research without the grant also, every study should have an ethical approval, irrespective of whether it is funded or not. So, and lastly, the institutional support or resources or the strength of the host place where you're going to work also matters. So these are the few points on which an application is usually reviewed by us. And most of the other funding agencies, they can have plus and minuses in the evaluation criteria, but this is how it is. So because we tend to forget the guidelines and apply to us as they wish, because they think the people who are reviewers or experts, they are some kind of ideal person. It means they will share our interests. I like to work in thrombosis or hematology, but maybe the person has an expertise in hematology, but he is not interested in thrombosis. So they might not have the same excitement about the proposal we are giving them to review. They are people from the scientific, life sciences, or medical field, but they might not be an exact expert of your proposal, because unfortunately we receive such a huge number of applications that it might not be very fair in terms of a reviewer who is looking into your study, because they might have some understanding, they might not have understanding of any some new technique. So there you can have a plus or a minus point from their side. They actually do not have time to read 105 pages of your proposal. This is a problem with most of the people who submit to us everything bulky. What do you think? You're trying to confuse the person who is reading it. People lose interest in your idea. So it has to be as crisp and sharp as possible. There has to be a proper title, not a confusing one. What you want to do should be reflected from your title. Similarly, very clear objectives and objective. It is a 3-year project, so maximum you should have 3 objectives. If somebody give me 9 objectives, I will just say it is too ambitious. Or you're trying to confuse. I will do characterization, that is 1. I will do validation, that is 2. No, all these things will go in one objective. So again, very clear and distinguished objectives need to be there. Similarly, review of literature or literature background doesn't have to be for 50 pages. Nobody is reading it, that it never happened or whatever. When you tell us what are you doing and why are you doing it, it will be sufficient to tell a person or to make them understand that this proposal or a study would be worth the time and the money. So, and again, you cannot expect every expert or a person who is reading your proposal, even if you give it to your supervisor, if there are 5 people, he or she is not going to be very impartial with every one of them. So there is always a human biasness when we judge something. Maybe it is a style of writing, maybe it is how the proposal has been formatted or served. So your dish should look temptable to people who are going to read it. So please be nice and be kind to your applications, whosoever is eligible. When I talk about SRF, RAs and postdocs, and similarly the MBBS MD students, but for the faculties, they will apply every year because this is for their career advancement. So ICMR is also planning, we will have the grant writing workshops for new people and what we expect from them. We will also try to help people who have given us a good idea, but maybe their project is not up to the mark. But we will still help them to modify with the help of the expert committee so that the end product is as desirable as we want it to be. So the last slide is the overview. Is do your homework. Homework means anyone who is a JRF, we leave the JRFs out because they will come from an entrance and they will start their career. But for other people who are already in the lab in some form, they need to do their homework before they start writing a proposal. It is just not key. There will be 5 compounds in a lab and each one will pick one and they will have the same proposal. Just replace the compound's name and that's it. It is not acceptable. Whatever you're going to work on, it should have a scientific background and a scientific future. It is at most important right now. Even there is a survey going from the Office of Principal Scientific Advisor to Government of India, and they have circulated a survey for scientists below or faculties below 45 years of age. And there also the first question is, what kind of research should India support? Should it be just theoretical or fundamental, or should it be something product oriented, which will give something to the society once it is done? Or it should be just numbers, the statistic analysis of something in terms of a number. So please think when you now apply to us. So whatever has been done, it was in the past. We will definitely have new guidelines for submission of an application to us, and there will be very, very rigorous screening of the same now. So this is just a message. The grant process is never wasted. It means if somebody is not successful with their application, it is a lesson. It might be disheartening, obviously, but it is a lesson that why my proposal was not accepted. So based on just like a publication or a research article where you got rejected comments, you will have comments or recommendations from the committee that if this was like this would have been better. So it is professionally fulfilling. Even if you have a not so successful proposal, it might be changed into one. It is just you need to focus your thoughts that what went wrong with it. So I would like to thank you, Professor Dash, Dr. Kamlesh, for having me here, and thank you for listening to me. I'm happy to take questions. Thank you. I think you're on mute.
Thank you, ma'am. Ma'am has been very elaborate on her talk, and as she has seen best of both the worlds, she has been a researcher, she has been an administrator. So she has told us how we can better our proposals, and I think both all the participants, including faculty and the budding researchers, all will be benefited by her talk, and will and way ahead before the research proposal submission. As she has elaborated that we should first clear our minds and be crisp and be targeted towards our research proposal, so that the organization as well as the reviewers will find it lucrative. So it was a very nice one of your presentations, ma'am. Thank you so much for your time, and I welcome all the participants for their questions. And I request all the participants to please introduce themselves and then ask their question.
Good morning, madam.
Good morning.
Madam, I am Dr. Christopher from All Indian Institute of Medical Science, Kalyani.
Okay.
Thank you for excellent presentation, ma'am. I mean, as a proposal, it was as crisp and straightforward.
Thank you so much.
Yeah, I have two questions. First thing is that when can we expect, like, is it throughout the year ICMR accept proposals, or do we have?
Right, since before 2017, when everything was offline, that is not digital, we were accepting proposals throughout the year. After that, now the proposals can only be applied through the portal, which opens once in a year. In last 3 years, it has opened around second week of December till second week of January for a month or so. So, but this year, as we have new DG and we have changes going on, so but it will open. But it right now there is no specific timeline when it will open, and but it will be once in a year for now.
Okay, and now as we are not, as ICMR is not sure for this year 2022, how frequently I should be checking the website, madam? Like, you give a timeline of 1 month. So what do you suggest? How frequently should I be checking for call for proposals?
Hello.
So you said if
Hello. There's a network issue, I guess.
To check whether the portal has been open or not, because it keeps on flashing that the portal is now open. Maybe this year we are going to develop guidelines, so maybe we will include for next year. Then when will this portal be open on a fixed timeline? For this year, what you might do is you can just check it once a day, and you check every funding organization. If you are a eligible candidate and you think you might apply to a program, then you should check it at least every 3 days everywhere. It's just not ICMR.
Okay.
For a postdoc fellows, if you apply outside India, people check it every hour because they are in need, because it opens. Their timeline is different and everything, so you don't know when it is going to. So right now, for last 3 years, it has been December, January cycle, but this year it might be little delayed or so, but it will come with a guideline. Hopefully, we are able to fix so that people have less inconvenience.
Okay. Thank you, ma'am. My second question is, now I have to investigate a parameter, and in my institute I don't have that facility,
Yes.
and I want to outsource that. So does ICMR have any particular criteria wherein which only government organization should be included for the collaboration?
Usually, any government organization prefers to have a tendering or an outsourcing given at government rate. So, if you are even doing it to a private person, they should give it on a government rate, because there is always, even for a test, let's say for next gen sequencing. So, there might be a lab from a government place which might be providing it at a lesser price, but they might not be adequate to support you. Instead, you go for a private player, but they have to match up with this particular number. So yes, you cannot actually just go to anyone and price a quote, because they check here that if something is being outsourced in a proposal, in that proposed budget, they check for it.
Is there a way that we can get that rate, ma'am? That how
The
much?
ICMR doesn't have it, but in your field, whatever, like if you talk about a clinical test, so if you're getting some blood parameters or something like that, then there is government approved rates. So
Okay.
that you can apply, get from any lab. So similarly, and your provider can be asked to give that particular number to you. So so whatever you test you plan to do, you just check at a government place where it is being done, just to check what number it comes out to be. There might not be a commercial lab, but they always know what is the rate of the kit or per test. So from there you can have a sample size that needs to actually, you cannot do without a private player. In that case, you need to know that number.
Thank you, ma'am. Thank you. Thank you so much.
Thank you, ma'am. We have a few questions in the chat box. If you can answer them.
Them. So, Dr. Anisha Matthew, she has asked that she wants to know if the travel grant will start anytime this year or next year for international conferences. Right now, because of the COVID restriction, it was suspended. Now we have new DG, so hopefully he will take a decision on this particular question, that whether they want to start this travel grant for non-ICMR scientists or students, and from which year he wants, from this year itself, because only 1.5 months remain, or maybe from next year, because right now it has been suspended on the COVID travel restrictions, and now he should take a decision whether they want to have it or they want to discontinue. They might discontinue also. It is just not the decision that it will start. It might be that it is just stop, because it's a matter of money. So I hope it answers your question. Then I have question from Miss Amina. Can a researcher apply for SRF having a career gap of 1 year after having 2 years? See, yes, You can apply for SRF if you are below, I think, 35 years of age, without any, because then we also apply the reservation as per government norm. You may apply, but for this you need to attach yourself to a lab. You need to be currently placed with someone. Yeah, yes, if you have MSc or M.Pharm plus 2 years, with a gap doesn't matter, and a publication, but currently you need to associate yourself with someone. You need to have a host, because independent SRF or RA doesn't mean you're independent. You have to have association with a person who is going to supervise you, a lab where you are going to work with. And she has also, she has, I think 2 times she has written the same. Yeah. These are the only questions, I think. Yeah, I have question from Vishnu Priya. Do we have any ICMR grant to pursue postdoc or research outside India? No. If you postdoc or research means a job, so you have to apply on your own and go there. Apart from that, Government of India do have collaboration or joint calls like Fulbright fellowships, where you can work for anywhere between 2 months to 1 year in a lab or so. But for that also you need to be associated with an organization in India. So those are the fellowships which will give you opportunity to have a research profile outside India, and there are many others, but they are not more than 1 year, and plus you will have to come back here, because you just represent India as the Indian ambassador, a cultural ambassador to those places, and to promote Indian culture there. Plus you can have the research proposal to you. That would be an extension of what you do in India. But if you completely want to go out and work as a postdoc or a researcher, then it is a job that should be paid from the country host where you want to go. And
we can invite few more questions. Yeah. If you have any few more questions, please feel free to ask or in the chat room.
yes. You may drop your message in the chat room, or you can ask me. It's fine.
Ma'am, from your talk, what I got is that now ICMR or other funding agencies as well are more on the translational part of the research, and in your research you also should have a thought in the first place that if this research is going to go for some positive results, then what will be the translational aspect of this particular research, isn't it?
Right. It's like now you cannot dump your project. Anyone. If somebody is saying I'm going to work on this and in 3 years I'm going to actually get a huge outcome or output, and what about it afterwards? I will just leave and go for a postdoc here. Just like that. No. You have to tell us what you are going to do with your thesis. This is just not ICMR. I think Government of India is changing the research policy based on the fact that we are piling up these degrees, or we have so many good faculties in India, and they are not performing as per their caliber. So maybe it is obviously there is a lot of restraint from an organization or a host itself. That is also one point to keep in mind. But with the research, government doesn't want to spend money on something which will not give anything after 3 years. Because when we talk about a publication, it is a personal gain.
Yeah.
For a fellow or a PI, it is a personal gain. Because you will say, I have this publication, I have so many citations, me, you, anyone. But rest what? With this study, how it is going to help the science, help the people, help the country. So this is what the new policy would focus on, that fundamental research is fine, but it should lead to somewhere positive. It doesn't mean that you will have a nature science or a patent, but and nothing comes after 3 years also. Everyone knows. Maybe you need a decade or so to come out with the product. But beyond that line, that I will continue to work on this project further. So you're getting what we want is, if you have a project, you should continue it. In what will be the next step? So this might end at 3 years, but I will do this after this. So you should be very clear. So you should not be very narrow in thought process कि 3 साल में 3 ऑब्जेक्टिव है खत्म. No. 3 के साल के बाद उन 3 ऑब्जेक्टिव से जो रिजल्ट आएगा वो उसका क्या करेंगे?
Right.
How will you sell your results? So that is one key point. So it is not the translational outcome or what is value of the project. It is that how it can be taken up further.
Yeah.
If you yourself or somebody else wants to pick up from here, how it can happen.
Yes. So one more question from Dr. Ibanella. I think she is also from A.M.S. Kalyani. That she wanted to know that there was a mention in your presentation regarding the publication and impact factor for applying a grant. So is this a proposed criteria, or this has been finalized, and how much should be the impact factor for?
Everybody is talking that how to this will be one I think a little harsh, but if the impact factor goes in the peer reviewed journal, not a predatory one, and it goes beyond 5, then we will be filtering out people who don't have an impact less than that. When we talk about an ad hoc project, or maybe a postdoctoral or a research associate. So it is more for the senior people.
Yeah.
Because,
Yeah, right. Like,
so this is one of the criteria, yes. So a high impact doesn't mean we will ask for 12 or 24, but there will be a number. A 5 may be a good number to start with. But it is, I am just telling that when we discuss, it is one of the point that how we can have eligibility criteria for someone to apply to us.
yes.
So if they show with this particular thing that they have a good publication in a good journal, so that would be a start. And again, you have a publication in something else and you propose to work in, this will be correlated. If
Yeah.
you have a good publication, it means you want to work in this field only. So if you are talking about thrombosis, you should show your career graph in that. That is how we will produce good scientists, not somebody who just for the sake of 3-year project jumps from plant biology to biotechnology to something else.
Yes.
So, and they cannot actually guide a good student also later, because themselves they are not sure in which field they want to work.
Yes.
No, citation doesn't. Citation is for you. The paper should have a good impact factor.
Okay. Thank you, ma'am. It was a very nice and elaborate presentation from your side. We are really thankful that you have sought time from your really busy schedule, and it has benefited most of us. This was our online workshop this year. We are definitely trying to make it offline next year. So I hope that you will grace this our next workshop with your present in Varanasi. So we thank you.
I want to just ask one question to Hinaji.
Yes,
Now your new DG has joined.
yes, sir.
Well, and how he visualizes to make the existing research scenario in the country or ICMR, or what he wants to bring about. What is your view actually?
Yes, sir. I was trying to tell his vision in bits and pieces, because we do not have a proper guidelines. So I cannot actually say anything with confirmation. But after having a talk, he thinks that we need to have more collaborative research. It should not be one-man army. You cannot be successful if you have, see, some researchers are rich. They might have a whole institute to them, but the new faculties or freshers, they might not have that kind of facility. And also, when you have an idea, it will have multiple components. Research is not one thing. It might have chemistry, it might have physics, some kind of math, some kind of life sciences, clinical, non-clinical, whatever it is. So you cannot, that is why I said one person cannot be expert of everything. So he is more interested in having a collaborative research proposal rather than one person saying I can do everything, which is not possible practically. So it is not about this is mine. It is about you have a very good proposal where you share your publications, share your patent, share your work so that it reaches to more people. You will be able to train more people in a better way if you have a collaborative work. So that is one of his vision, and second one is you should have cutting edge topics when you apply to ICMR. So it should not just restrict to something very archaic, very old phenomenon which is known, and you cannot possibly have an answer. So what? I want to do this, but so after that, so what? So if you are writing to us, it should answer by itself that if I do this, after 1 year it will give me this, after 3 years this will be the final result. Based on what it can be taken up further, or I would like to take it up further. So this particular flow chart needs to be very clear when next time somebody applies to it, because right now he is kind enough to give us time and he is reading each and every file. So we will also have to change our perspective about how to evaluate a project. But from his point of view, the same, as one person asked me, they want to go out and do postdoc or research. If that person does very good job there in some other country, why this person cannot be given an opportunity here to work on similar grounds? So it might be something too idealistic, but if we try, we may be able to change our perception, because our IITians, our researchers, they go outside and they bring so many laurels, but in India they are not able to do so. So why they are not able to do so, we have to ponder over it and work towards to keep them here, so that they just don't want to go to a Western world and perform. Why can't they perform here? So all the ethics and the genuinity we need to inculcate in our younger people here only. So what he thinks, and accordingly he wants people uh, to apply to us based on those guidelines. And for us, it is our responsibility to choose certain people. Their project might not be perfect, but we will help them to make it close to perfect, so that we are sure of what they are going to bring at the end of it. It might be no result. It is fine. That's a risk. But we want to take that risk with certain people. So this year the number will usually go down when we talk about selections. But those, all of the people or the fellows who get selected, we will help them in modifying what they want to do for the betterment of the results that we expect.
Okay, thank you, ma'am. Thank you for being here. And now we'll proceed with our next presentation on this confocal microscopy. Thank you, ma'am. Thank you for being here.
Yes. Thank you, sir. Thank you so much. Have a good day.
Yeah.
Welcome all. Our next lecture is by Dr. Shruti Mishra. She is Assistant Professor in Bone Marrow Transplant and Stem Cell Research Center in I.M.S. B.H.U. She is having 17 research publications, including blood and International Journal of Lab Hematology. So, start the volume.
Speak with you all today. And I know that it has been almost an end to the 4 days long scientific and intellectual feast, and you must all have got a lot of points to take back home with you. So without further ado, I will delve into the topic of today, that is monitoring antithrombotic therapy. Now, this topic I will be discussing in 3 different parts. In the first part, we will discuss what is the basics of normal hemostasis and what happens in thrombotic disorders. In the second part, we will discuss about the antithrombotic drugs and their mechanisms of actions. And in the last part, we will see what are the various newer modalities and newer methods and newer sites of research in this antithrombotic therapy region. Okay. So normal hemostasis is defined as a method where the blood is to be in the liquid state when the vascular system is intact, and they should be converting into a gel-like form whenever there is a breach in the continuity of the vascular surface. Now, what happens initially whenever there is a breach? Because of reflex mechanism, there is vasoconstriction and platelet activation. Along with that, there is activation of the coagulation cascade and the formation of blood clot happens. Now, this platelet activation and vasoconstriction are primarily considered as the primary hemostasis. The coagulation pathway is primarily considered as a secondary hemostasis, and the antithrombotic and fibrinolytic mechanisms are also considered as tertiary hemostasis. Now, we know that coagulation or hemostasis for that matter is a double-edged sword. Too much of anything can be problematic. If there is clotting within intact blood vessels, we get thrombotic patients. If there is no clotting, whenever there is a breach in the continuity, then we have the bleeding disorder patients. So too much in anything, it's not good for the thing. Now, for the basics of hemostasis, we have to know a bit about the physiology of the platelets. Now, what are this? We all know that they are anucleated discoid structures. Now, where do they come from? They come from megakaryocytes. The first structure or the first identifiable precursor of megakaryocytes, which are identifiable in the bone marrow, are the megakaryoblasts. And they are induced by TPO or thrombopoietin, which will give rise to the maturation of megakaryoblasts into megakaryocytes. Now, there is a basic difference between the maturation of megakaryocytes from the other lineages. Now, what is that? The thing is that the mechanism by which megakaryocytes mature is called endomitosis. Now, what is endomitosis? Normally, what happens in mitosis, we get 2 daughter cells. That means whenever the nuclear material is doubled, it is equally divided into the daughter cells. And in humans, what we get is a diploid, 2N in the daughter cells. But what happens in endomitosis is that the nuclear material is getting doubled, but there is no division or there is no formation of daughter cells. So the haploidy of the megakaryocytes is usually polyploidal. So normally a megakaryocytes will be ranging from a 16N to even a 64N. Okay, so it is a polyploidal cell. And since there is endomitosis is happening, megakaryocytes are the largest cell which are identifiable in the bone marrow topology. Now, how do platelets come from them? If you see the topography, normally megakaryocytes will be seen lining the sinuses. Now, why is that? Because mature platelets are pinched out from the cytoplasm of the megakaryocytes containing the granules, that is the alpha granules and the dense granules. They form this, they throw these pseudopods through microtubule formations, and there are proplatelets formed. And these proplatelets give rise to mature platelets, which is directly released into the circulation through the sinuses. Now, this is the morphology that we see in the bone marrow. These are the megakaryoplasts, which are very large cells. These are the immature megakaryocytes, which have still got some bluish content in the cytoplasm. And these are the mature megakaryocytes containing granules, which are normally present in the platelets. Okay. Now a bit about the history. The first person to ever describe platelets was Max Schulz in 1865. He was a German anatomist. In 1881, Bizzozero, an Italian pathologist, correctly determined that platelets are involved in hemostasis and thrombosis. In 1886, William Osler found that platelets have a significant role in the thrombus generation in atherosclerotic disorders. In 1906, an American pathologist, James Wright, established that megakaryocytes are the cells which are giving rise to platelets. Also, interestingly, John Wright also found the staining method that is called the Wright stain, which is a type of a Romanowski stain, which helps us to visualize the cell lines. Now, platelets, they are anucleated structures. So there are some similarities between platelets and RBCs. Both are anucleated. They have some. They have this cytoskeleton under the cytoplasmic membrane. But the major difference between platelets and RBCs is the canalicular system. Now, this canalicular system is also connecting the interior of the platelet to the outer surface. Now, why it is important? Because when the platelets are getting activated and the granules have to release their content, there this canalicular system comes into action. This is an electron microscopic picture of platelets, and you can see that underneath the platelet membrane, there is an extensive skeleton, and there are a lot of actin filaments. Now, these actin filaments are the one which helps in platelet retraction and clot contraction after the clot has formed, and these are the zigzag membranes. And the platelet surface has got glycophospholipids and the receptors, GP1B9, and also integrins like alpha-2B, beta-3, etc. Now, what happens whenever there is an injury? As I have told initially, whenever there is a breach in continuity, the endothelium just besides the site of injury will release an hormone called endothelin. Now, this will cause reflex vasoconstriction at the site of injury. Now, why is that? So this will prevent the loss of blood naturally. Now, along with that, there is also release of certain other proteins, and there is exposure of the collagen. And collagen is one of the strongest activator of platelets that is present. Now, what happens in the primary hemostasis is that the platelets get activated and then they adhere to the underlying collagen. Initially, what happens through GP1B2A, the platelets will directly adhere to the collagen, but it is a very weak bond and they will be washed off because of the shear force of the blood. Then what happens? The von Willebrand factors are meanwhile activated and they will come and bind on one side to the collagen, and on the other side, it is going to bind with the platelets via GP1B59. Because of the shear force, there will be rolling of the platelets on the collagen surface. But since this bond is very strong, the platelet do not wash off and they tether to the underground subcollagen surface. And along with that, there is activation of glycoprotein 6 and further activation of integrins alpha 2 beta 1, which gives rise to strong adhesion of the platelets on the subendothelial collagen. So basically the same thing that we have seen in inflammation, just like neutrophils have a rolling on the surface of the endothelium. Similarly, platelets also have a rolling mechanism on the subcollagen, and initially it is a weak bond and later on it becomes a strong bond and the platelets adhere to the subendothelial collagen. Along with adhesion, there is activation of the platelets. They throw pseudopods, which increases the surface area, and there is release of the platelet contents, and the alpha granules and dense granules contents are being released, and this gives rise to aggregation. What is aggregation? Where platelets attach with each other, not with the subendothelial collagen. They are now attaching with each other to cover the damaged area, and this attachment happens via fibrin through the receptors glycoprotein 2B3A. So obviously, meanwhile, coagulation has started and fibrin generation has begun for this aggregation to happen. Now, what are the contents of platelet granules? The dense granules have mainly got a lot of agonists like ADP, ATP, serotonin, and calcium. Alpha granules have got various different contains like adhesive proteins like fibrinogen, fibronectin, von Willebrand factor, growth modulators like platelet-derived growth factors, TGF-beta, platelet factor 4, coagulation factors, most important, platelets are the most important source of factor 5, and they are also source of fibrinogen, factor 11, high molecular kininogen, and also antithrombotic factors like factor protein ACE and plasminogen activated inhibitors 1. Now, whenever the platelets are getting activated, these granules will be, through the pathway of GTP activation, they will be binding with the surface through syntax and SNAP23 and release their granule content to the circulation. So basically, the functions of platelet is for adhesion to the subendothelial matrix, aggregation between each other, secretion of the platelet granules and proteins, and induction of the procoagulant activity. Now comes the secondary hemostasis. What happens in secondary hemostasis? We are talking about the coagulation pathway. Now we all are familiar with this coagulation cascade or the waterfall model, where there are two paths, extrinsic and the intrinsic pathway. Extrinsic pathway, there is the activation of tissue factor and then activation of factor 7 to 7A. Factor 7 then activates factor 10 along with factor 5. Together 5 and 7 activates factor 10 to 10A. 10A will activate prothrombin into thrombin, and thrombin will release fibrin from fibrinogen. Okay. Now what happens in intrinsic factor is that factor 12 along with prekallikrein and high molecular kininogen will be activated to factor 12A. 12A will activate factor 11 to 11A. 11A will activate factor 9 to 9A. 9A will activate factor 8 to 8A. 8A along with 5A will activate factor 10, and then so on and so forth. And there are certain lab tests like PTINR, which will test the extrinsic pathway as well as the common pathway. ABTT tests the intrinsic pathway and the common pathway, and thrombin time tests only the common pathway. Okay, now there are certain drawbacks with this model. Now, what are the drawbacks? Drawbacks are the H-man factor or factor 13 does not cause bleeding in most of the patients. Factor 11 deficiency causes variable symptoms, and factor 8 and 9 deficiencies like in hemophilia A and B causes severe bleeding, even though it causes severe bleeding. Okay. So now we know that there are certain drawbacks in the waterfall cascade. So nowadays we all follow a protocol called the cell-based model of coagulation. Now, what is the cell on which this coagulation is happening? This is nothing but the platelet. Now, what happens when there is a platelet activation? There is also activation of enzymes called flippase, floppase, and scramblase. So, when these enzymes are activated, phosphatidylserine, which is normally lining the inner leaflet of the platelet membrane, will now be flopped on the other side, and it will be now exposed to the external surface. Now, this phosphatidylserine is the strong base on which all the coagulation factors go and adhere and gets activated, and all the coagulation pathway happens on the surface of this platelet. So this is called the cell-based model of coagulation. Now, it has also got 3 steps: initiation, amplification, and propagation. Now, what happens in initiation is that because of injury, there is release of tissue factor, and this tiny amount of tissue factor will activate factor 7, 9, and 10. Now, this factor 9 and 10 will activate factor prothrombin and give rise to a trace amount of thrombin. Now, this pathway, you will say, why tissue factor is not then giving rise to the further coagulation pathway? Say, for example, like we have said, the doubt over there, factor 5 and factor 8 and 9 deficiencies are having severe bleeding, while the extrinsic pathway is functioning properly. So the answer lies here. What happens is this trace amount of thrombin that is being generated will not only activate downstream factors, but it will also activate tissue factor pathway inhibitor. Now, because this tissue factor pathway inhibitor is being activated, this mechanism of thrombin generation stops here itself. But this trace amount of thrombin is very important. Why is that? Because it now goes on to the second step of amplification. What it does is that it activates factor 8 and factor 5 to 8A and 5A. And it also activates factor 11. Now this factor 11 gives rise to amplification of activation of factor 9 to 9A. This amplification is quite variable in nature. Because of that, we have seen patients with factor 11 deficiency, they also can have variable presentation. They may present with severe bleeding, they may not have any symptoms at all, because this amplification is also variable. So this answers our second question. Now, after this amplification pathway, so what exactly happens is the formation of the tennis complex and the prothrombinase complex. The initial tennis complex that was there, it was the extrinsic pathway tennis complex. If you want to talk about in respect to the waterfall cascade pathway, this is the more sturdy or more prolonged activation complex, which is called the intrinsic tennis complex. So what is this complex made up of? It is made up of factor 9A, activated factor 9 and activated factor 8, which then binds factor 10 on the surface and cleaves it, gives rise to activated factor 10. This activated factor 10 will move from the tennis complex and then go and bind with 5A to form the prothrombinase complex. The prothrombinase complex will now bind prothrombin or factor 2 and then cleave it to give rise to activated prothrombin. And then once you have the activated prothrombin, activated thrombin, it is giving rise to fibrin from fibrinogen. So this thrombin is more sustained and more amplified than the initial trace amount of thrombin that is being generated. But still, that initial trace amount is very important for the amplification and the propagation to happen. So this method and this cell-based theory of coagulation answers all the three questions that we have, that why factor 12 or the H1 factor deficiency does not have any effect on the patient. That means this patient do not present with any bleeding at all. Why factor 11 has got a variable role, and why factor 8 and 9 are so important, and why the deficiency of this patient's presence with severe hemophilia. And we also learned from this method of cell-based technology that cells helps localize the clot at the site of injury. So it doesn't happen on the surface of inactivated platelets. It happens only on the surface of activated platelets, which are there at the site of injury. So your clot is being formed at the site of injury and not anywhere and everywhere throughout the circulation. Coming to the last part, thrombus and antithrombotic events. There is release of tissue plasminogen activators, which has got fibrinolytic activities and thrombomodulin. So basically what happens whenever there is whenever thrombin is being generated, it will act on endothelium to give rise to tissue plasminogen activator. Now this plasminogen activator will give rise to activated plasmin from plasminogen, and plasmin basically is a fibrin cutter. So it will degrade fibrin, whether it is in the normal circulation or it is a part of the clot, into fibrin degraded products and D-dimer. And there are other factors like heparin-like molecules which binds with antithrombin, and this complex inhibits factors 9A and 10A. Whenever thrombin binds with the thrombin receptors and thrombomodulin is released, it will activate protein C. Along with protein S, they will inhibit factor 5A and 8A. Also, activated endothelium releases prostaglandin I2, nitric oxide, and adenosine diphosphate, which inhibits platelet aggregations. So these are the antithrombotic and antifibrinolytic effects of the endothelium. Thrombin is the most important factor which is being generated by this clotting mechanism. It has got multiple functions. Not only it induces clot formation, it has got effects on the antithrombotic and fibrinolytic effects, and it has got an inflammatory actions by inducing platelet adhesion, neutrophil adhesion, monocyte activation, and it has got a role in healing by inducing muscle generation by platelet-derived growth factor generation. Okay. So basically, hemostatic system is a fine balance between this procoagulant and anticoagulants that are being synthesized in the body, and tilt of balance to any side will give rise to various disorders. Like more procoagulants, you will have thrombotic disorders, more of anticoagulants, you can have fibrinolytic disorders and bleeding disorders. Okay, so now this finishes the first part. So let's go see what happens in the disease entity. So in thrombosis, like Workout said, there it is a triad or three mechanisms which give rise to thrombosis. Either there has to be some endothelial injury. Either it is because of hypercholesterol or inflammation. Hypercoagulability has to be there. It can be either inherited like factor V Leiden or protein C, protein S deficiency, antithrombin deficiency, or it can be some acquired hypercoagulability like in cases of disseminated cancer. Blood flow has to be abnormal. There is that means there is a loss of the laminar flow, either there is a stasis or there is turbulence. Stasis because of prolonged bed rest, turbulence because of atherosclerotic vessel narrowing, etc. There are various risk factors which gives rise to thrombosis. The shared risk factors are old age, elevated blood mass, body mass index, smoking, family history of myocardial infarction, thrombophilia, stasis, inflammation, OCPs or hormonal replacement therapies, etc. For arterial, usually the metabolic syndromes like hypertension, diabetes, elevated cholesterol, obesity are the risk factors. For venous thrombosis, immobility, prolonged bed rest, major trauma, surgeries, cancers, pregnancy, long distance travel, sitting in one place for a long time, like in long flights, frequent flights can give rise to thrombosis. Now, these are the various screening tests which are done. Basically, we do PT, APTT, and then we have to, for ruling out antiphospholipid antibody, we have to do DRVVT, incubated APTT mixing test. For individual deficiencies, we can do assays, protein C, proteinase assays, and we can do functional assays as well as the antigenic assays. Factor V Leiden is usually assayed by activated protein C resistance. And then we can do genetic assays for prothrombin, G2210A, and homocysteine, etc. I'm not going into details of this because we are going to talk about the antithrombotic drugs mainly. So what are the indications of these drugs? Basically, all the thrombotic events like coronary arterial disease, cerebrovascular diseases, coronary artery bypass, AMI, stroke, transient ischemic attacks, all these patients are given antithrombotic drugs. Now, this is a slide which I have taken from ASH. So this is just showing what are the various points or the various sites where these drugs are active. Cell surface activation inhibitors are those which inhibit P2Y12 receptors or ADP receptors on the platelet surface. PAR1 inhibitor, PAR1 is PAR is the receptor for thrombin, which is present on platelet surface. PAR1 inhibitors are there like Vorapax. P2Y12 inhibitors like clopidogrel, prasugrel, ticagrelor, and cangrelor. Intracellular signaling by COX inhibition by aspirin and PD inhibitors like dipyridamole and cilostazol can be there. And platelet aggregation can be inhibited by directly inhibiting the surface receptor, that is 2B3A, that is by abscesimab, tirofiban, and eptifibatide. This is the same thing which we have discussed just in a diagrammatic way, where you can see the cell surface receptors, P2Y12 inhibitors, and these are the PAR inhibitors like Vorapax. Thromboxane generation are inhibited by aspirin, and then PDE2 inhibits formation of AMP from cyclic AMP, and this GP2-3A receptors prevents platelet aggregation. Now, there are various drugs which are being still under the trial, like those which are inhibitors for glycoprotein 6 antagonists like Rivacept, antagonist for GP1B95, that is anfibatide. This will prevent the adhesion. Then PAR1 inhibitors like atoxapa and Vorapaxa, and PAR4 inhibitors are both PAR inhibitors like bivalirudin are under use as it is. Then we have thromboxane A receptor inhibitors like ifetroban, which are still under evaluation. Now, this is just the same thing in a bit detailed manner. I am just going through this because I want to tell you that these drugs some drugs are direct acting and some are pro drugs. That means they are orally ingested and they have to be activated by the cytochrome mechanism in the liver. And once the active metabolite is formed, they will inhibit the P2Y12 receptor on the platelet surface. Now, why is this receptor important? This receptor is important because it activates cyclic AMP. Cyclic AMP, what it does is that cyclic AMP, when it causes phosphorylation of VASP, it leads to platelet inactivation. Okay, so that is what we are targeting to do. So these drugs are basically causing phosphorylation of VASP, and that will lead to platelet inactivation through this mechanism. This is just a busy slide where we are just showing all the various drugs along with the mechanism of action. And this is to highlight that drugs may be given individually, but they can also be given in combination, like aspirin and clopidogrel can be given together to patients. Or various mechanism of actions like aspirin and abciximab or clopidogrel and eptifibatide, these drugs can be given in combination. How do we test whenever we are given a drug to a particular patient? So there are various modalities. We can either do a platelet functional assay to see whether the platelets are having a hindered function. The basic modalities are light transmission aggregometry, verify now or whole blood impedance aggregometry, platelet function analyzers, platelet works, VASP assay done by flow cytometry, and thromboelastography and rotating. I will just describe slightly some of the most important modalities, or which are the commonly done in most of the labs. So most commonly what everybody does is light transmission aggregometry. So here you set the baseline with the patient's own platelet poor plasma. Then along with the platelet rich plasma, you add the agonist and see whether the platelets are clotting or not, aggregating or not. Okay. And there is a magnet over here which will keep this is basically an iron rod, and there is a magnetic field in the machine which will keep the platelets in circulation and prevent their abnormal aggregation and settling by gravity. What are the normal agonists which are used? ADP, epinephrine, collagen, TRAP, that is thrombin receptor activating peptide, arachidonic acid, and ristrosidone. These are commonly used. So LTA mainly allows us quantification of the final common pathway. Okay. So this is a semi-quantitative test. Whole blood impedance is basically through electrode methods where the platelets are initially in circulation. Once you add the agonist, the platelet will clot around the electrode, inhibiting the flow of electricity. That means there will be impedance which is happening. Okay, and measurement, we do it by ohm per minute and measure the area under the curve to see what is the functionality of the platelets. Platelet function analyzers are beautiful machines. They basically replicate what is happening in vivo, in vitro. So you have some cassettes which are lined by collagen, and there is an aperture, and the blood is pushed through that aperture at a high shear force. And this collagen is either lined by epinephrine or ADP. And we measure the time taken by the blood to clot, to prevent or close the aperture. Okay, so this is basically a point of care test, and very small quantity of citrated whole blood is required for doing this test. But the main drawback is it's very costly. And one patient will require one cassette and the cost goes very high. Okay, now VASP, as we have already mentioned. So VASP is activated by the P2Y12 receptor, and phosphorylated forms inhibits the platelet. So basically we are trying to indirectly measure by measuring if phosphorylated form of VASP is increased, that is vasodilator stimulated phosphoprotein is increased. That means it is an indirect measurement that your receptor is being properly inhibited by the clopidogrel drug that is being taken by the patient. So what is the method? We take the whole blood, mix it with PGE1 only or along with ADP PGE1, incubate it at room temperature, mix with permeabilizers and add fluorescent antibodies against phosphorylated VASP and do a flow cytometric technique. Using the flow cytometric technique, we measure the platelet reactivity index. So naturally, it is understandable that lower the PRI, better the compliance with the drug intake and the better the functionality of the drug that is happening for that particular patient. So VAS basically gives us an indirect measurement of the P2Y12 activity. And main profitable thing is you do not require a pre-treatment value to monitor the patients by using this technology. But the drawback is it does not correlate exactly with the platelet aggregation per se. Okay, so this is just a trace. These are the traces of LTA basically. So you can see that with ADP initially there was a aggregation that was happening, but then it was lost. And with arachidonic acid, there was no clotting at all. So this shows that the patient has got adequate intake of clopidogrel and aspirin, and the closure time with on PFA 200 was also prolonged with collagen epinephrine and collagen ADP respectively, and the PRI was low. So the patient was taking both aspirin and clopidogrel, and both are functioning, drugs are working well. Excuse me. Coming to thromboelastography and rotem, both are complementary. So what happens is that in TEG, the cup is rotating and the pin is fixed, and in rotem, the cup is fixed and the pin is rotating. It's basically a method to detect the clotting system and fibrinolysis system in the whole blood itself. The plots are quite complementary or similar in nature. In rotem, you get CT, that is the clotting time, and R, which is the reaction time in TEG. So this is basically the time taken from the initiation of the process to the time when the first clot formation is detected. CFT is the clot formation time, the time it takes to get up to 50% of strength of the total clot that is going to form. Alpha angle is same in both, which is basically showing you the kinetics of the clot formation. MCF is maximum clot firmness or maximum amplitude intake, which will show the maximum strength of the clot that is being formed. And then lastly, we also see the lysis in this patient. So LY30 means lysis at 30 minutes, LY60 is lysis at 60 minutes. And this is a CT is what is the nomenclature in rotem. So this will give you the clot stability. Now, why this TEG and rotem is important is that we can understand what all factor deficiencies or what all products we need to give to patients. This has got a multiple usage, but the role of TEG and rotem in monitoring thrombotic drugs has still not been dwelled into much, and it is a point where research can be done whether TEG and rotem can help us in monitoring antiplatelet and antithrombotic drugs. So we know that clotting time and R time are dependent upon the coagulation factors, whether the patient is having on any anticoagulants, high dose of high FDPs like in DIC patients can give rise to prolonged CT and R time. And then kinetics and CFT will be dependent upon the coagulation factors, platelets, fibrinogen level, as well as anticoagulant level. Clot strength will depend upon the platelets and fibrinogen and factor 13. And the lysis will all obviously depend upon the fibrinolytic enzymes and factor 13. Okay. So this is basically a point of care test for bleeding risk, but the data on potential assessment of this antithrombotic drugs is still limited. Other markers which we can do for platelet activations is the granular release, which can be done by flow cytometry or ELISA based. Circulating aggregates can also be done, but major disadvantage that these are like you can activate the platelets while taking the blood sample itself, and there is a lack of data, whether there is a clinical and lab correlation, the data is still lacking over there. We can also do a metabolite assay for aspirin. Serum thromboxane B2 measurement is still the gold standard to understand the aspirin effect on the patient. Platelet turnover can also be assessed by doing flow cytometry and doing ribonucleic acid. Now coming to anticoagulants. So initially when a patient presents to you with thrombotic events, initially IV heparin is given, and oral rivaroxaban, apixaban, and subcutaneous low molecular weight heparin and fondaparinux is given. For maintenance, IV dosage is converted to oral warfarin, and subcutaneous may be continued, but fondaparinux is still an off-level for maintenance therapy. Now, the names may look very strange and very difficult to understand. This is a simple mechanism by which you can remember the names. Those who are targeting activated factor 10A will have 10A in the name, like apixaban, endoxaban, rivaroxaban, fondaparinux. Those who are targeting 2A has got a B in the name, like agatroban, bivalirudin, dabigatran. Okay. Parin will be there with both 10 and 2A are inhibited, like heparin, enoxaparin, and dalteparin. Okay. So these are the basic mechanism of actions. So heparins and low molecular weight heparins will inhibit factor 2A, factor 10A. Direct factor 10A inhibitors are also there, like rivaroxaban, and vitamin K antagonist is your warfarin. So heparin, just like the one which we have mentioned in the normal hemostasis, remember the slide where I have showed normal heparin-like molecules are synthesized, they bind with antithrombin, and then this will give rise to inhibition of factor 2A and 10A. So this is the similar mechanism which happens when you are giving heparin from outside. That means you are giving a drug. Warfarin basically is a vitamin K inhibitor. So it will inhibit all the vitamin K dependent factors, that is 2, 7, 9, 10, as well as protein C and protein S. How do we assess or how do we monitor warfarin? We do a PT-INR because the pharmacokinetic is very narrow, and the too much of warfarin, slightly increased dose can give rise to bleeding in those patients. So we have to give them we have to tightly monitor them by doing regular PT-INR. For direct oral anticoagulants, either APTT or thrombin or icaritin or anti-10A direct quantification can also be done for assessment of their adherence and to monitor these drugs basically. There are certain other drugs which are given in acute conditions. When there is suddenly a patient throws a clot, what do? You give a clot lysis like streptokinase or ontiplase. What they do is that they directly melt away the clot by inducing activating plasmin, which will cleave the fibrin. So this thrombolytics can be non-fibrin dependent or fibrin specific. Fibrin specific are those which are directly activating the tissue plasminogen activators like altiplase, retiplase, and tenecteplase, or the indirect ones like streptokinase, urokinase, and antistreptilase. Now this finishes the drugs and their mechanism of action. A few slides, just a couple of slides more regarding the research points which can be future targeted therapy for us. So there is a concept called coat platelets now. So what are these coat platelets? They are seen in thrombotic patients, like they are called procoagulant platelets, where the contents of the alpha granules are released and they are now lining the surface of the platelets. So these are collagen and thrombin which are lining the surface of the platelets. So it is called as coat platelets. So they're activated platelets and they have a higher tendency to form a thrombus than non-coated platelets. So basically what happens is that intracellular calcium increases and there is activation of GP6 and the PAR receptors, leading to phosphatidylserine being exposed on the outer surface and formation of thrombus or clot. There has been studies which have shown that there is a spatiotemporal association between various factors and receptors with the clot formation. Like for the initial 2 minutes, the tissue factor thrombin and the glycoprotein 6 are more important, while in the later part for the thrombus to build and become more stabilized and more strong, PAR14 and integrin alpha 2 beta 3 is more important. Certain other studies which has been done on tetraspanin membrane proteins have shown that ADAM10 interacts with this and they have got a crucial role in activation and expression of GP6 on the surface. SPAN15 is also interacts with Rho GTPase and modulates the expression of GP6 surface proteins. So this all can be the targets for various antithrombotic drugs. There has been spectacular work done from the biochemistry department under Dr. Das' guidance. This has been a study which is published this year itself in Blood Advances, where they have found that the sonic hedgehog, which is a molecule which mainly is associated in embryogenesis, can be produced in the platelets from pre-existing mRNAs once they are activated. And this has got an important role in further downstream activation of GTPase-RAS and phosphorylation of myosin light chain. They also saw that if you inhibit this sonic hedgehog production, there is a inhibition in platelet aggregation, granular secretions, and overall platelet functions. So they suggested that it can be a future potential target for drug development. Similarly, they had another spectacular publication in Hematologica this year itself, where they have found that mitochondrial ATP is indispensable for platelet aggregation and procoagulant activity. And it is crucial for sustaining platelet granule secretion, for maintaining the platelet neutrophil interactions, and for thrombus generation. And they have found that targeting this can be very important in specifically in drugs for thromboinflammatory diseases like acute myocardial infarction, ischemic stroke, DVTs, and pulmonary embolism. So the take-home message from this talk is that there are still a lot of areas of research. Monitoring is not still perfected yet. We don't know still how to perfectly monitor antiplatelet and what is the role of platelet function analyzers and platelet function tests on antiplatelet therapy. And extensive studies can also be done on various other molecular mechanisms to get targeted therapy. Say, for example, GP1 alpha and 1 milligram factor A1 complex inhibitors can help us in developing a novel therapeutics for those patients who are suffering from immune thrombocytopenia. So this is it. These are some of my references. Thanks for your attention and listening to me. Thank you.
Yeah. Thank you all of you for patient listening. Our speaker is traveling and that is why this was a recorded lecture from her. If you have any questions, please ask us on the chat box, and then we will get back to you on your email for the elaborate answers. Yeah. After this session, we'll be having a session for application of confocal microscopy and its online demonstration by Dr. Nitesh Singh, who is a scientist at our center for I welcome Dr. Nitish. Nitish, please come in front. इनका कुछ क्वेश्चन नहीं था जो भेजेंगे No, it's mute kar do. Okay.
Ready? So, very good afternoon. Today we will discuss about application of confocal microscopy. This will be the last session for 4-day workshop, and I hope you enjoyed all those previous sessions. And in the near future, if you are continue to do that kind of workshop, we will definitely contact you peoples as we have your email ID with us. So
I have a few instructions for all of you. First of all, I request all of you to mail us again for your correct name because I think that few of you have given us the handwritten applications, and because of that there might be a spelling mistake or something like that in your certificate. So I just request you to again mail your name and designation and institute to us. Uh, just write a mail, don't attach anything, so that we can again send all of you your certificates with your correct name and designation. That is the first request. Secondly, we'll be sending you the email for SERB, who is the sponsor of this workshop. Please write them to write it to them for positive feedbacks about this workshop, so that all those who have worked tirelessly for this workshop will get a due recognition at the sponsoring agency, and again will be able to have online offline sessions for this kind of activity, and you'll be able to come to us for that as well. So I'll email all of you regarding this workshop, this email, and I request all of you to mail it to them. So that was the second request. Okay, Nitesh, please continue with your.
Confocal microscopy, also known as confocal laser scanning microscopy or laser scanning microscopy, and it is an optical imaging technique for increasing optical resolution and contrast of micrograph by means of using special pinhole to block the unwanted or unfocused light in the final image formation. It can be also known as that using a confocal laser light to control the depth of the field and a pinhole to eliminate out of focus light, thus allowing visualization of image in a single horizontal plane. So this is an 3D image of microtubule in a plant protoplast. Next. Okay, to understand the confocal microscopy, here is a ray diagram. So this is a laser system, and which will emit the laser light, and this will pass through the confocal pinhole, and it will now focus to the sample in a focal point. Okay, so after irradiation with laser, the light will be emitted from the sample, and it will goes to the detector by passing through the pinhole. So this will allow us to avoid or remove the unwanted or unwanted light from the sample. So this is a laser light. It pass through the pinhole and strike the sample, excite the sample at different focal plane. This will give you information of the cell at different focal plane, which will be detected by the detector and form the image to the software or computer. So to better understand the confocal microscopy, let us watch a video. So in a normal fluorescent microscope, we have a fluorescent unit which will emit the sample with laser light or fluorescent light. So that is how the sample will be radiated with fluorescent light, which will emit the emissions from the fluorescent antibody present within the sample. So that is how light will be emitted and detected by the detector. So here we will get a blur image because the sample will be formed image of unwanted light which will pass through the mirror to the detector. But in case of confocal microscopy, we have pinhole. The light passes through the pinhole and strike the sample or irradiate the sample with laser, and this will strike at the focal point so that unwanted light will be not emitted from the sample, and again it goes back to the detector by passing through the pinhole. So while moving the stage at a particular direction or degree, we can have 3D image by focusing at different focal plane or at different focal depth of the sample. So at last we will have a 3D image by using confocal microscopy. Okay. So that is how we get a 3D image of a sample. Next, we have Zeiss LSM 700 confocal microscope with us, and this is a pinhole system. We generally omit the unwanted light from the sample or or from the laser system, and it has one halogen system for bright field microscopy. This one is a halogen system. This is a TPMT used for DIC imaging, and we use ZenBlack software with that machine. And there is one laser system below that table which have four different laser, uh, belongs to 405 nanometer, 488 nanometer, 555, and 639 nanometer. So according to your sample or fluorochrome, you can use different laser with your samples. So difference between wide field and confocal microscopy. As wide field absorb light from the or detect light from the various unfocal plane, so that will have a blur image. So these are the bright field images, and these are the confocal image which have a crisp and clear images. So bright field microscope have a wide light area which will be detected by the detector. So at last we will get a blur image. So this is also a wide field image, and in compared to that we have a confocal crisp image. So another application of a confocal microscopy is a tile scale microscopy, where you can scan a whole organism. If it consists of a larger size, in that case you can divide the sample in a sample or your organism in different tiles, floor tiles. So this is an image of a zebra fish, and this is a Drosophila larva. So here the image is divided into different tiles, and at last the final image will be formed by merging all the tiles, and you have a good looking picture at the last by merging all the images. So this is the one of the application of the confocal microscopy. Another aspect or another application of confocal microscopy is a live cell imaging, where you can image live cells by using, if you want to image cells for longer duration, for that you need a CO2 incubator facility with the confocal microscope, where you will provide 5% of CO2 continuously to the cells. So in that way you can image live cells. So here in this video, I'm going to show you how macrophages engulf cancer cells. Here cancer cells are stained with the red color, and macrophages are engulfing them or clearing them. So that is how we can use confocal microscopy for lifestyle imaging. Next, for light calcium imaging, we can also use confocal microscopy. For that, we just fabricate a slide, and that slide was coated with polylysine, and cells were put on the slides and allow it to adhere to the base of the slide. And after incubation for 5 to 10 minutes to adhere the slide to the microscopic slide. So light cell imaging, we use the flow antibody for light calcium imaging. So for cell having low calcium, we will have some blue kind of image. As calcium increase, there is a red color in the image. So this is the control or vehicle. So as you can see, there is very less calcium present within the cell, and this is a positive control 83187, that is called as ionophore. That is called ionophore, which creates pore on the cell plasma membrane and allows the entry of calcium from outside to the inside of the cell. And there you can see a red dot and yellow dot, which shows there is increase in light calcium within the cell. So in this work, we also see the effect of prion protein on platelets. For this, we incubate prion protein along with calcium. So how platelet will get activated by prion protein? So it generally cause entry of the calcium from outside to the inside the cell by using trypsin channels, which are present with on the plasma membrane. So as you can see, there is more red, means there is more calcium entry and more platelet activation. So for checking that whether these calciums are coming from out of the cell or from outer area of the cell, so we chelate the extracellular calcium with EGTA, and again we add PRP or prion protein, then we find there is no such calcium rise by adding prion protein. So that means prion causes calcium entry and causes platelet activation. So this is a kind of work we have done with confocal microscopy. Another aspect of confocal microscopy is Z stacking, where we can make a 3D model of a cell. Suppose this is a cell which will divide into the different focal plane, and from one plane to another plane. So we can have a 3D image by dividing a cell into different focal plane. So this is a image of a pollen grain, which is imaged by the confocal microscopy. So why we use Z stacking? To study the presence of a desired protein or some desired drug. So whether it is present within a cell or diffuses within the cell or present or bound to the outer membrane or plasma membrane of the cell, or whether it goes to the nucleus. For this, we can utilize Z stacking. Suppose the protein present within bound to the outer area of the cell. So you can see there is more fluorescence at the outer or outer focal plane. This is the outer focal plane. If the sample present within the or to the nucleus or within the cell, uh, we will have fluorescence in that area. As it is present on the outer membrane, so we will have hollow region in the center focal plane. So with this experiment, we can understand that the protein will bind the plasma membrane or goes into the cell. Okay, so how to perform the Z stacking? For first of all, we have to first set first and set last and set first on the software. For this, you have to first focus the cell where it begins to visualize at the microscope. Then set it first and make it out of focus in another direction. So you will have a certain micrometer depth, and just start the experiment. Then you will have get a a 3D image of a sample or a cell. So I have a question. Can you tell that this with this video where we incubate where we incubate A beta, which is stained with dye. So when we incubate this with our platelet, it is engulfed by the cell. So can you tell where it is present, where this prion protein is present? It bound to the cell membrane or present within the cell? So please type on chat box. Okay, we will have that in the later on session. So please type that answer of that question. Okay, next approach we can use for confocal microscopy is a fluorescence recovery after photobleaching. It generally used to determine the kinetics of diffusion through the cell or tissue, or to find out the active components of cell or recycling of the components. So to better understand the frag, we can have a video of a frag. इसको बंद कर दीजिएगा ना। नहीं, ये वाला माइक बंद कर दीजिए, मैं ये वाला ऑन कर लूंगा। No. इसको बंद कर। Here we So to understand the lateral mobility of cell membrane protein, we generally stain membrane protein with GFP. So this is a GFP tagged protein on the cell membrane, and we put 100% laser light to bleach that area by using a computer guided laser. So that is how we bleach the area on the cell membrane. Okay, if there is a diffusion between the protein, there will be recovery of the fluorescence at the bleached area. Okay, so in case of cell, if we selected the area on the cell and bleached to see that there is a diffusion or a mobility of protein on that area, we can find out after bleaching, there is a recovery of the fluorescent in that area that shows there is a diffusion of protein. Suppose that protein is firmly anchored with the cell, so then we bleach the area, but we did not get the fluorescence at that bleached area. So that means there is no diffusion, or that protein is firmly anchored, so there is no diffusion. Okay. So Next, to better understand this wrap, we also perform some experiment with wrap. For this, we need a fabricated slide where we form clot by using fibrinogen, thrombin, and calcium chloride. So for that, we need a fabricated slide. First of all, we form a clot within that fabricated slide and allow it to form for 30 minutes incubation. Here we use fluorescent labeled fibrinogen, and after forming a clot, we just focus that area, that is a 10x image. And after focusing, we will have a single thread of a fibrinogen or fibrin clot. Then we select this area on the that fibrin thread and expose to 100% laser to bleach that area. If there is clot lysis or there is mobility on that fibrin, there will be recovery. So when we compare, then we check the recovery of the fluorescence. So there is a normal recovery graph. So you can see the control sample, there is very slow recovery. When we treat the sample or we, as we use TPA or streptokinase of drug, there will be fast recovery. That means there is molecular dynamism present within the clot, or that is clot lysis occur. So that is a bleached area. That is how we bleach the area, and there will be recovery in that area. So we will better understand by after this session, we will understand the online demonstration of the confocal microscope. So at last, this paper was published in Blood Advances, and that also cover page was published by them, and this image was taken by our group, and they published that image on their cover page, and that is the paper. This is the paper published in Blood Advances. So that is a big achievement for our lab. Thank you for attention, and the dice is open for questions. Please. If you have any question, please ask.
In chat box
Unmute है सब लोग. Any questions regarding confocal microscopy, you can ask, or you can mail us also. We will ready to solve your queries regarding microscopy. Sir, I think there is no questions. We can move forward.
Please forward with the present online presentation.
video
अच्छा हाँ video हाँ. Next, we will have a online demonstration of confocal microscopy. So we will better understand how it works and how this software support at various techniques. If you have any question, you can type in the chat box. We will answer the questions.
Hello, this is Ivan Lahadem from A.M.S. Kalyani.
Yes, ma'am.
In that FRAP experiment, no, what samples did you take exactly? I don't understand. Is it like?
Yeah, we use fibrin clot in that experiment.
Okay, so you expose the clot to laser, and then you see how it again recovers or something
Yeah.
like that?
So suppose if there is any clot lysis, we will have faster recovery in the FRAP experiment.
Uh-huh.
Okay. If clot is very firm, then we will have very slow recovery after photobleaching. Suppose we add some TPA or streptokinase to the clot, we will have fast recovery as compared to the control sample. So that is a whole thing about that experiment.
Okay. And what about that Z stacking? Is it like you take like the platelet cells, or and then you do like a depth scanning
Yeah.
or something?
So in case of G-stacking, suppose we find out we want to find out whether our desired protein goes into the cell or into the nucleus or certainly bound to the plasma membrane. So in that case, you have to perform Z-stacking to find out that our desired protein is goes into the cell or bound on the cell membrane. So
For that one, no, that you've done. I mean, shouldn't nucleus also be stained so that you know whether it goes to the nucleus or not? Because I can see
yeah.
only the red stain.
So in that case, you just also stain the nucleus and another stain for your desired protein. Then you will have a merged image of both nucleus staining and also that your desired protein. And finally, you will get the image that your desired protein goes into the nucleus.
Okay. Okay. Thank you.
Yes, ma'am. Okay. So next we will have an online demonstration of confocal microscopy where you can better understand how it works and the different application of it. ये लास्ट वाली देखो क्या है शेयर हो रहा है क्या सर वीडियो कौन सा है सर क्या वीडियो वो जो वो जो आपके इस पे है ना हाँ I'm going to demonstrate the preparation of microscope slightly for the use of the confocal fluorescence microscopy. So before starting, I'm going to show what are the requirements for the preparation of slides. So first, you can see here. Building a simple box in this we have placed some tissues are which are treated with the water and we need fibrates 2.5 micrometer, 100 micrometer fibrate and 1 ml of fibrate. One marker pad, 1x CPS, which are used to wash the slides. And this is the slides which at which which are used for the settings. This is the slide and we also need the tritone. And we need tritone to permeabilize the cells and we need 4% for paraformaldehyde to fix the cells. We need the 2% acid to block the solutions. Block the state and we need so how we going to start the experiment? So first we take slides. And the specialty of this slide is we do not have to put the slide with the polymerizing because this slide is already coated with some chemical agent. That is why we do not need to fix with the polymerizing. So this slide has an upper layer, upper side and lower side, and here is some here we can observe and sign by which we can detect the upper side and lower side. So in the lower side, we just mark the patterns at a particular area where our nested cells. Let's see the boundary. At this boundary, we place our nested cell here. At this slide, we place into the box. And take 1 ml of water. Pour the surrounded area of this slide. This slide can drive. Next, so after placing this slide, we just add cells. So we are taking the platelet. Concentration 1 into 10 to the power 6 per ml. So we take 100 microliter of platelet sample and place into this selected area. And this platelet getting adhered to the surface, and this will take approximately 15 to 20 minutes. So we leave the site for 15 to 20 minutes to complete this box. And we cover this box with the aluminum foil because during the course of our experiment, we do not want to any light to enter into our slide area because there is in the further step we are using the fluorescent antibody that is light sensitive. That's why we are covering cell. So after 20 minutes of incubation, we need to fix this slide in the paraformaldehyde. So we are using 2% paraformaldehyde, and we have to take equal volume of the paraformaldehyde as I take the volume of cell. So we just take this slide will these samples were fixed after 10 minutes. So it will take 10 minutes to fix all the samples. So just wait for 10 minutes. After 10 minutes of incubation with the paraformaldehyde, we need to wash this slide with the 1x KBS, and we for the next step we are going to formalize all the cells which are adhered to the slides. So before formalization, we have to wash this slide with 1x KBS for 3 times. Like this. So here a node cell will be removed from the slide. Now this slide were formalized with the 0.01% triton, which are suspended in 1x KBS. We have to add 100 microliter of this volume, and this will incubate it for 2 minutes. So after 2 minutes of incubation with the triton extracted, we just wash this slide again for 3 times. Remove extra triton detergent. So here the tritonX will be removed from the slide, so it will not affect the cells and antibody to be used in the experiment. Now, after the permeabilization, I'm going to block the cells which are adhered to the slide. So for the block, we are using the 2% PSA section, which are solubilized in 1% PS, 1x PS. So, in 100 to 200 microgram of 2% PSA on the slide. And it is made for the 15 minutes for complete blockage of non-specific site of percolates, non-specific blockage, non-specific percolation which are created by trichome. So we are permeabilizing the cells because we are using binding up primary antibody at their particular sites. So we close this box and place it into the 4 degree temperature. And shaking is this incubation no need for the shaking. So we just basically just place it with the 4 degree. Say the 4 degree temperature. Secondly, everybody will become pregnant. So we have so after the overnight incubation of this slide with the primary antibody, first we have to wash this slide with the Onyx fluids 3 times. Okay. this was can remove all the excess antibody which are white, which are not bounded form. After see, we have to incubate this slide with the fluorescent tagged antibody. So I'm using Alexa Flow 488 anti-rabbit IgG, and this antibody we are using in concentration of 1:200 in dilution in 1x previous with 2% PSA. So in the next step, I'm going to say, so because this antibody is fluorescent tag, so we need to turn off lights. Then, so I'm taking the secondary antibody. One is to 489 dilution. It's into the 2% PSA. This antibody solution was taken 100 microgram based on the slide. Tested area. And this slide would be incubated for 1 hour at room temperature in dark rooms. And after the incubation, we have to wash this slide and cover with the cover slip and with the glue, and then go for the image. So after 1 hour of the incubation, first we have to wash this slide with 1x EPS 3 times. And after washing, I just place this slides in the dry tissue paper to dry this slide, because in presence of water, our image is not good. So we just dry it for 2 to 3 minutes. So now our slide is ready to fix for the imaging, and before imaging, and I'm going to address this slide with the cover. This slide with the cover slip, and to fix this cover slide, I'm using anti-fading agent, which are act as the adhesive agent. Take a small drop of anti-fading agent. This cover slip. See the cover slip, and this will be placed on our sample area. Now this slide is ready for imaging. So now I'm going for the image at confocal microscope. So this is solution system. So this is a pinhole and mirror unit. This one, and this is below one is a laser system, and this is a supercomputer which is used for this microscope. And this is a panel from which we will set 10x 20x objective lens, and this is a fluorescent unit to acquire images or to scan the images during microscopy. Okay. This one is an integrated microscope which one is going to use, and we are going to use a Venplex software for this for microscope. Okay, so before start the system, the Venplex system asks for start system, which will boot or check the system configuration before going to start any experiments. Okay. So we will align the laser system and mirror before starting any software. Okay. So we will acquire the images in fluorescent mode first, then we will move to the mode. So Agla, create the slide on the microscope. So we will put the slides upside down, meaning the cover sheet will face the microscope sideward. Okay, and properly will hold it on the microscope stage, and then we will okay, wrong line. Then select the FITC filter and on this. So now we are focusing the design area in the slide to see the platelet structure of actin. So after focusing on 10x, we will move to confocal software.
Angle change.
Select that for Alexa for radiated. So adjust the
ऑडियंस को क्या दिख रहा है?
So we are now focusing the slide on by using 488 and by setting gain or by increasing or decreasing. We will view the potential of the cells. So these images we have taken at 10x, and each dot represent here the fluorescent green color represent a single cell, single platelet cell. Okay, so this one is a fluorescence image, and this one is a DX, and this one is a mark image. Okay. Nobody is able to see. Now we are moving to the 48 or 63x of the microscope. For this, we will use So due to fluorescent microscope, we have to switch off the light. So because of that, we are not getting such a good quality of video. So please kindly consider this. Now we are focusing the slide on 63x. Slowly increase the gain. Range indicator will describe. Okay. So these are the platelets. At 63 years. Okay. So click on the range indicator to check the gain setting. Click on this range indicator. Okay. So if we increase too much gain, this will increase the gain. Increase the gain setting. So if we increase too much gain, we have to check that how much we should acquire the gain setting. So for that, we have to select on range indicator. Then accordingly, we will select the gain master setting. Okay. So again, click on the range indicator after setting the gain. settings. You can clearly see the nice picture of platelets, which is stated by what is the stain? Okay. Now we will see the different channels for excitation, the confocal. So we will use now slide which is having tissue. Okay. So first of all, we will see the 633 excitation we have, and we have 568, we have 488, and this TPMT is a and this is a dummy. Okay. So we will first require event and set all the gain setting for each individual excitation. So for that, we have to select all channel DIC from here, and after selecting all channel DIC, we will go to individual excitation to select the clear images. Okay. Okay. Now we are going to like it. So after selecting excitation of 633, we will adjust the gain setting at this excitation. Okay, here this is good. And uncheck this one, and again we will select another excitation, that is 568. So we are getting like this. So this is also good. Okay. Next, uncheck it and select 488 excitation. So it will feed the computer to align or excite the laser corresponding to 488 excitation. So this is a 488 excitation. Okay. After setting the gain and the setting, and this one is the DAPI, which generally signal is here. This is the DAPI. So here we are as the tissues is having some autofluorescence. So here we do not need any kind of fluorescent antibody to stain the tissue. So we just use this one to have all kind of excitation. So that's why we are using a tissue sample which have autofluorescence. It gives fluorescence in every excitation. So after selecting all those channels, we will image. This one is 633, and this one is 568, and this is a 488, and this is a DIC, and this one is a TAPI, and this one is a Mars image of all. Okay, so this in 2D view, we will have this kind of image. So this one is a 633 excitation. This one is a 568, and this is a 488, and this one is a DIC image, and this one is a DAPI, and this one is a merged image. So here we are not getting any kind of signal in 568. So for this, we will just increase the laser. For this, we have to stop and uncheck all to get the laser head setting for this particular excitation. ये वाला इसके कारण हो नहीं है इसको मैं okay. So because of close excitation, we are not getting any fluorescence in that region. So we will just increase the distance between the collision of the point of excitation of different filters. So after merging this, we will get this one image. Okay. So having images at different antibody tag to the tissues, we will acquire images, and after having different images, we will get the merge image to find out the coincidence of or presence of two different protein at the same place, or where the proteins will present at the different site, whichever protein of our interest to study. So this is one kind of study. Next, we will see the tile scan to scan the larger area on the slide. Suppose we want to scan a zebrafish or want to scan a whole Drosophila. So as, for that, we have to make a slide of that Drosophila or zebrafish. Then we will use a tile scan setting to acquire the whole image. So for tile scan, select the desired area and check the tile scan tab. After checking the tile scan, it will calculate the size of the tile that will be 5 into 5 and just start the experiment. So for that, we will uncheck the DIC setting. So we will have a good tile scan to have a good image. Just we will start. So you can see the whole image divided into different sections. That is one tile, that is another tile. So that image will acquire at the different time to acquire the large section or a larger slide. To acquire Drosophila or to acquire zebrafish, we will use tile setting to scan the whole organism in that manner. So you can see we will have different tile sections. You can imagine, suppose we are putting some Drosophila or zebrafish in that particular slide, we will have a different region in the tile scan, and we can acquire the whole organism in a single slide. So let's first complete the whole scan. We will move to the another application of the confocal microscope. The slide is just for a demo purpose, so we are not doing some experiment kind of thing. We are just informing you people about the application of the microscopy, how we can use this machine in our different kinds of experiments. So after completing tile scan, we will just check how to do Z stacking to scan different section or layers of a single cell to find out the presence of our desired protein, which is present within the cell or present extracellular to the plasma membrane or to the surface of the cell. Suppose if you want to check whether our desired protein or some kind of desired protein which will go into the nucleus, to find out that we have to go for Z stacking that will give information about the presence of that desired protein which is present on the surface of the cell or to the core of the cell. So for that we are using Z stacking. So here we just show how we perform the Z stacking. So we will use the same tissue slide to show how we can perform the Z stacking. This will give how much slice of that particular area it will take to perform Z stacking. We have to first select the desired area or desired cell. So here you can see that there is taking a block where you have to select the range of the slices from the first image to the last image. And for this, you have to first you have to select the Z stacking. Okay, then uh, light the image. Okay, just set unfocus the image to select the last or the first, whichever you want to choose. So this one, this could be our last, and after then move to the other side to unfocus that area, and after getting a little bit of that reason, then check it for the first. Now you give the computer or the software to give command that you have to take image at different slices. Suppose you have a ball and you will image that ball in a different section. So selecting the first and last of the Z stacking, just click on the start experiment. So this will have around 176 slices of that current focused area. So just start the experiment, and here you will have the remaining time and the percentage of skinning yet to be done. Okay. So after getting all images from Z stacking, this is a platelet state. So here you can see the staining is getting intense when we move further from outer to the end region. So here we will get more fluorescence in the center. So this implicates that our interested protein or region or desired protein is present within the cell. Suppose if that protein is this video I'm going to demonstrate section. So here you can see to the outer area. So this is a statistic of the previous slide. So you can see as we move we will get more fluorescence in the cell. So this tells us that our protein is present within the cell. Okay, so we can also check. This is a this is another fluorescent. Here we use the 488 hexafluor antibody, and in a 3D image, these cells look like this. Okay. So this is also a 3D image of another cell. So from this we can tell the presence of our protein. Now we will discuss about next application, that is fluorescent recovery after fluorescent bleaching. So here in our previous experiment, we use fluorescent labeled fibrinogens to get a fluorescent fibrin clot. So this is a clot. This is a fibrin clot which is scanned by 488 labeled antibody that bind to the fibrin clot. So after selecting a desired thin fragments of the fibrin clot or thin thread of fibrin, we will just like this. We have a fibrin strand here. We will select a region of interest where we will perform a bleaching and a reference where we can use that region as a reference and a black region for reference. So after performing fluorescence bleaching by using 100% laser at that specific region, we will have bleached area in that region. So if there is a more dynamism of the molecules, that fluorescence recovery will occur fast. Or if we have low molecular dynamism, we will have low recovery at that region. So in that graph, from this graph we can tell that there is a recovery of fluorescence in that region. Okay, this is that image where graph is shown. As already discussed in the lecture of the confocal, you can clearly see how we perform the FRAC. Okay, so this is all about the FRAC. If you have some doubt, please tell us. Thank you.
So now dice is open for the question. Please ask if you have any doubts and questions. Please let us know. Unmute कर दीजिए. Unmute. If you have any doubt regarding confocal microscopy, you can ask us, or we will move to the next session. We will send you a workshop form. Feedback form. Feedback form. Yeah. We will move to the next session now. Camera.
नीरज जी को बुला दें क्या? हाँ सर, नीरज जी सर को अगर बैठे हुए नीरज सर, हाँ बुलाइए, सब वैलिडेटर के लिए इंजेक्ट करेंगे. नीरज जी को किसी प्लेस प्रोवाइड देते हैं जी.
So here we come to the last valedictory session of this workshop. I thank all the participants who have enthusiastically participated in the workshop, and I'm very thankful to you that you have taken your time and make our efforts fruitful. Now we'll start with the valedictory session. I will briefly introduce everybody and then we will start with the valedictory session. Soon after this valedictory session, we will be sending you the feedback form, and also will be sending you the email for sending the feedback, so that all of you can first of all, I will request Professor S.P. Mishra, sir, head of our department, to say a few words to the participants. Sir.
So, good afternoon, everybody. So, I think this workshop gives immense pleasure to our. That means, in case of thrombosis and platelet, and novel things you learn here, and especially COVID-19. What are the effect of COVID-19 on platelet and these things? So I think very fruitful, and wish you all to in this workshop. So thank you very much.
Sir. Okay. Now I will request Professor Das, sir, coordinator of this workshop, to say a few words to the participants.
So now we are coming to an end. It is a 4 days of intensive workout on both the sides, from our side as well as from your side also. Quite a bit intellectual exercise. I think the take-home lesson would be that there is a emerging. It's not an emerging area for in our country. We can say that it is an emerging area of platelets and thrombosis biology. It's a wonderful area, and you can start working in this area, and to go ahead with the work. I think this has given a window to your stimulus trigger for your thought. But on your side, those who are really serious to have a academic professional career in this area, I would advise them to first read. You can read papers. You can read papers from our lab method section. We are very elaborate, used to be elaborate. But you can read many more papers to understand, especially platelet preparation part and what they wanted to do. Recent papers, I suppose, and so that you can find out what are unexplored areas. Because if you do research, you have to go for unexplored areas. So you have to yourself find out. Everybody's thought process is different from another person's. And there is a saying that chance comes to the prepared mind. Chance or serendipity. That divine spark of an idea will cross the mind of a person if the mind is well prepared, well cultivated. So well prepared means you have to give enough time, real seriousness for the study. And as I told you that platelets in India, hardly any competition. All the platelet experts, plateletologists, or whatever term you use, you have met in the cyberspace in the 4 days. There is no institution, another center for platelets except ours, unlike cancer biology, etc. So you will find a pretty area of competition, less of competition. So the basic premise of this workshop was that to have more and more people get interested in this area of research. And I am very much sure that it would have invoked thought process in your mind. Any difficulty, we are here, but it is your career. You have to work harder, and we can contribute less, but still we are ready to contribute whenever you want some help from us. Don't get panic away from the equipment gadgets you have seen, because these gadgets here in our department is a 30-40 years endeavor. And if you start from scratch today with seriousness, whatever customized equipments you need, you can gather, or you take collaborations with other labs, referral laboratories, by making some payment. If you have a seriousness, money will come, no worry at all. So with this, I wish you all the good luck.
Thank you, sir. Now, I would request one of the participants to please speak their mind so that we can get a real-time feedback on from your side. Dr. Mahesh, Dr. Ibenella, Dr. Dr. Thomas. Yeah. Yeah, please introduce yourself and come.
So I'm Dr. Anisha Matthew from Lady Harding Medical College, New Delhi.
Yes, ma'am.
So these 4 days have been very informative because platelet signaling and even its role in COVID and all became much more clear. And you have actually opened a new path for us to study more, not just in COVID, but also in other conditions. So I thank you, sir, for holding these 4 days, especially with the practical aspects also and introducing us to how we can start from the basic level and reach to where you're currently now. Thank you so much, sir.
Thank you, Dr. Anisha. We are happy that our workshop was of use to you, and definitely it will guide you. If you need any help, definitely you can email to us. Now you have our email also, and please keep in touch. And if you explore this area more and more, our center is the only center in the country, so definitely we'll be happy to help you out. Thank you.
Thank you so much, sir.
Anybody
Hello,
else?
this is
Good afternoon.
Yeah. Yes, ma'am. Who was it? Yeah.
hello, sir. Actually, I want to thank you so much for for this program and all that. I got like a last minute information on it, but I'm very, very happy that I got to join this workshop and I got to know a lot of things. All the lectures were very well delivered, very informative, and it keeps me, gets me thinking on some research ideas or something like that. So I thank you immensely. Thank Professor Dash and the team for organizing such a workshop.
You are from I.M.S. Kalyani?
Yes, sir. I'm from I.M.S. Kalyani.
Okay, so
I'm working.
Student, head of the biochemistry from he's a MD pass out.
Yes, I got the information from him actually regarding the workshop.
One of our best products from our department. Okay.
Yes, sir. Yeah, I mean, we're very happy to have him as a HOD. I mean, he has a lot of ideas and all that. So, very happy.
Thank you, ma'am.
Good afternoon, everyone.
Good afternoon, sir.
This is again from Kalyani, Dr. Christopher.
Yeah, Dr. Christopher, go ahead.
Yeah, sir, at the outset, thank you so much for putting this new idea into my brain, because my work was basically with respect to CNS and other psychiatric disorders. So my whole idea of attending this workshop is not exactly, to be very honest, not with respect to thrombus biology, but there are a lot of overlapping cardiovascular disorders, comorbidities with respect to psychiatric patients. So that was my point, and I am sure that I have got some spark in me, like, which kept me thinking, and I'm sure that something would definitely come out of it. But I sincerely request your help in modifying the abstract, or with respect to methodologies, whether is it possible or whether it is too ambitious for a new institute like this to have. I request your support also. And I thank all the organizers. It was wonderful, fantastic. Thank you so much.
Thank you, sir.
Hello, sir.
Yes, sir.
This is Mahesh from University of Hyderabad.
Yes, sir.
First of all, I'm very much thankful to everyone, and especially who introduced me a lot about this platelet biology. And coming to my work, actually, I used to work on megakaryocyte biology. So after this, interaction and all, I understood, so many techniques and different approach like netosis and, platelet biology in other dimensions. And so in final, I want to tell, thanks to everyone. In next session also, I'm very enthusiastic participate in the next session. Thanks so much. Yeah.
Thank you, Dr. Thank you, Mahesh. Few of the participants were really enthusiastic, including Mahesh, and that is what the response that keeps us going. And I must thank all of you for your interesting questions to many of the presenters, and also we will definitely welcome all of your newer ideas, because these newer and newer ideas are the things that keep us exploring on this field. Thank you. And I must thank SCRB and ICMR, our sponsors, for this workshop, because without their help, we will not be able to go for this workshop again and again. So I request all the participants to write it to SCRB regarding if they have any good reviews for this workshop. Definitely, I hope that you have. So please write it to SCRB through email that we will be providing us soon.
Sir, one last thing. This is again Christopher here. If we need, if I need an observership there, to see the lab and how to do the platelet extraction, what are your institutional rules, if you may say so?
Please send us a request through proper channel, and we will see what we can do from our end.
Okay, thank you so much, sir.
Anything, Josna, you want to speak a line? So these are our small faculty, Professor Misra, head of the department, myself, Kamlesh, who is always with you. Now he's Abhijit from our department also. And Josna has presented, she's sitting here but out of focus, but she has presented flow cytometry data to you. And a very small department, but happy to say very committed people. She is Josna now. Hi, bol dijiye. Okay. So very committed people. So so now what is the next? Now we should conclude. Or?
Yes, sir. So I thank all of you for attending this workshop again, and we'll be concluding here. And I again request all of you to send your name in writing and also institution, so that we can dispatch the certificates at the earliest. Thank you, all of you, and I hope that when we'll be floating the application for the next offline workshop, many of you will be interested to come here and join us. Thank you. Thank you. Thank you again. So now we'll be concluding the session.
Thank you. Telemedicine के जितने लोग
And last but not least, I must thank Professor N.K. Agrawal sir and all the staff in the telemedicine, Shashi ji and Abhijit and all others who have really helped us to carry out this workshop without a small glitch, and their facility is wonderful. And I also thank all our non-teaching staff, including Rajendra ji, Akhilesh, and Umesh, and all our students who have helped us to carry this workshop. Thank you.
Signing off.